Welcome back. Yeah,
You need to start from one day.
can you all hear me?
Yes.
Yes.
Please mute your mic who are not participants.
I request now Professor Prahlad K. Seth, PK Seth sir, and Professor Raj D. Mehra madam to take over the dais. And I also request Professor Raj D. Mehra madam to introduce Professor Prahlad K. Seth sir and Professor PK Seth sir to introduce Professor Raj Mehra. And then I request the chairperson, Professor PK Seth sir, to introduce the speaker and Professor Raj Mehra madam to tell about the lecture. Like PK sir had told that you can decide. Please, over to Raj Mehra madam and PK Seth sir.
Thank you.
Und ist es in der Pfanne oder im Marmeltopf, ne?
Shall I start? Anita, shall I start?
Yes. Yes, Dr. Mehra, please.
In the session.
Yeah, this is always the problem with the online conference.
Yes, I know.
So can I help you somehow here? Just tell what I have to do.
Can you, am I audible?
Yeah, Dr. Mehra, you can go ahead.
Now, we have a keynote lecture starting. Professor Suresh Ratan from Aarhus University, Aarhus, Denmark. Speak. And we have
Is it outside?
here.
Can others unmute, please?
Whom we his main affiliation is with NASI. He is the Senior Scientist, Platinum Jubilee Fellow, and Distinguished Scientist, Biotech Park, Lucknow. He is the Chairman, ILSI India, and former Director of the IITR Lucknow. Specific research interest of Professor Seth are various projects on neuroscience, biotechnology, and toxicology has been his very sort of very close area of research. Professor Seth has received many honors. Just to name a few here, he has
Can I
one of the.
Can I interrupt for a second? Madam, you please switch on your video.
It was not coming. Okay. So shall I go ahead now?
Yeah, please
Yes,
continue.
you were audible, but we could not see you. Yes.
Okay.
Now we can see, madam. Madam, we can see now.
Okay. So Professor Seth is one of the founders actually. They have founded it from the Lucknow, the IAN, past president of the FONS, Acharya PC, he has got Acharya PC Ray Gold Medal of ISCA and BK Bachawat Memorial Lifetime Award of Indian Academy of Neurosciences, and is the distinguished toxicologist of Society of Toxicology, is the fellow of the major national science academies of the country and maybe other countries too. So Professor Seth will now.
Thank you. Thank you. Thank you, Professor Mehra.
Professor Suresh Ratan.
For this elaborate.
Over to you, sir.
Yeah, thank you, Professor Mehra, for this elaborate introduction. And I have a pleasant duty before I introduce the speaker. I have to introduce you Professor Raj Mehra, whom I have known for at least 3 decades, and we have worked very closely for the Indian Academy of Neurosciences and in the field of neurosciences in general. Dr. Mehra is associated with the Department of Anatomy of the All India Institute of Medical Sciences, New Delhi, and her research interests are in the field of the neuroinflammation and neurodegeneration. She has published a number of research papers, of original research papers, and has contributed a lot to the field of the neurosciences. And thank you, Dr. Mehra. And now I have a pleasant duty to introduce today's the keynote speaker, Dr. Suresh Ratan. He's from the Department of Molecular Biology, Genetics at the Aarhus University in Denmark. And Dr. Suresh Ratan's interests are in the field of aging, health, and longevity. He has been bestowed with several awards. I'll just mention a few. He's the founder and editor-in-chief of the Journal of Biogerontology, which is published since 2000 by the Springer Nature. He's also chairman of the biological section of the International Association of Gerontology and Genetics European region since 2013, which he is continuing today. He has been awarded with prestigious Lord Cohen Medal and also Wiley Birmingham Medal. Writer of several books for experts, general public, and children. And today he has chosen the topic of his keynote lecture is very interesting, and that is aging research, fact, fiction, and fantasy. With this, I request Professor Suresh Ratan to kindly deliver his keynote lecture, and later on, Dr. Mehra would like to make any comments. Kindly go ahead, Professor Suresh Ratna. Over to you. Yes, sir. Namaste.
Namaste. Can we?
Can you unmute, please?
About 2 years
Yeah.
ago, one of the main organizers of this Indian Academy of Neurosciences conference, Professor Anita Jagota, invited me to give a keynote lecture at this conference. I was very much looking forward to meeting you all in Hyderabad. At that time, we never knew what will be happening in 2020. So now, courtesy Corona, we are all sitting in our own lockdown place and trying to communicate with
It's
each
not audible. Kindly arrange the...
Little louder, sir.
participate in your 38th annual conference of the Indian Academy of Neurosciences. And I will be talking for about half an hour, giving you an overview of the field of aging. Because within aging field, this role of the brain and neurosciences is very, very crucial. So I will just give a general understanding of what we know in the field of aging, what we want to do with that knowledge, how much we can actually do, and how much is just our wishful thinking or fantasy. So when we talk about aging, there are basically two ways of talking. One is aging as if it happens throughout life, any change occurring with the time. Some people would like to call that as aging. That's why in language, we often use the word, how old is your baby? Yeah? My granddaughter is now 4 years old. Is that what the word aging we are talking about here? Perhaps not. Yeah? In the linear scale, that yes, aging can be considered any change happening with the time, but that does not make distinction between the way we understand the word when we use the word aging and old age. And that is the second way of looking at that, aging and old age as a different stage in life. or different stages of life, and one of the stages is when aging we talk about. Because when we talk about the word, or we use the word aging, it's not the child which comes into mind. What comes to mind is this kind of phenotype, that aging is the period and the process during which our bodies become progressively weaker and weaker. And we become more and more frail. There are more chances
Please mute who are
of disease,
not speaking.
eventual death. So that is the common understanding of the word aging and old age. So the first part, the left part here, is not the aging part. The downhill part is the aging part. And when does it happen? During our lifetime? In evolutionary terms, especially in biology, what do we think? When does it happen? It happens if an organism lives longer than its essential lifespan. Now, this is a new term in the field, introduced in the field of aging, to compare with what we know maximum lifespan or average lifespan, but there is something called essential lifespan. Essential lifespan means the lifetime needed in evolutionary conditions for the evolution to fulfill the purpose of life of a biological system. So the purpose of life of a biological C. elegans, Rectus rectus, Drosophila melanogaster. It can sound very depressing. That is that the purpose of life of me? Yes, as a Homo sapiens, as a member of the Homo sapiens species, that is the purpose of life. As a human being, our purposes can range from beautiful art, literature, philosophy, religion, whatever we want to talk about. But today we are not going to talk about that. So essential lifespan is the lifespan needed by a species in its natural environments to grow, mature, and reproduce. Like Drosophila in its natural environment needs about 7 days to do this job. But when I bring the Drosophila in the laboratory, and those of you who also work on that kind of model system, it lives 1 month, 2 months, 3 months. A rat in nature, the essential lifespan of a rat is about 1 year. But in the laboratory, it can live 3 years. Same thing is about Homo sapiens. Our essential lifespan, the way nature requires it, nature means evolutionary processes, evolutionary selection, that is not more than 45 years. But now we know that in laboratory conditions, which means highly protected conditions of modern societies, we can expect to live double, or somebody has lived almost triple than the essential lifespan. So aging, when we talk about aging, aging sets in if we live longer than the species lifespan. That is when aging sets in. Yeah? So ELS is the time, as I already mentioned, that aging sets in after essential lifespan, and the essential lifespan is this definition, the Darwinian definition of essential lifespan. Now, why evolution has allowed aging to happen? Why evolution has not made immortal systems? There are a wide range of theories, very complex and beautiful theories by evolutionary biologists given. And I don't have time to go through them, but there is generally this, that is an evolutionary neglect, because in nature, not many organisms actually survive beyond essential lifespan, or it's a genetic dustbin, or accumulation of mutations, or antagonistic pleiotropy, or disposable soma theory. These are fascinating, beautiful theories developed by the evolutionary biologists, which tell us that evolution has not selected for immortality. It allows aging to happen. And that is when we now it will come whether it allows aging to happen or it causes aging. That will be coming soon to that question. So during the last 50, 60 years, hundreds of biogerontologists like myself and all around the world. There have been good tradition of aging research in India also. That's where I was originally introduced to aging research. We have described what happens during aging at almost all level of organization. This is a great achievement, right from the population level to individual to group, individual organs. That's where the brain people have done tremendous contribution in understanding what happens to the brain and its all components and the cells and the molecules during aging. And then going down from the organs, systems, the cells, then there's a huge area of the metabolome where there is correlations made that during aging, this happens, this happens. I have just highlighted some of the main areas which are often talked about these days. What happens to stress responses? What happened to DNA methylation? What happened to telomeres, glycans, mTOR, IGF, every molecular pathways? It's a huge area. You are all aware of that. And that's not a small achievement, showing that if we live longer than our species lifespan, if we live beyond essential lifespan, which is a great achievement of ourselves because evolution only wanted us for about 45 years, then during that period, one or more or all of these changes will happen. But most importantly, these changes will not happen equally to any two people or even within the same body. No two parts of the body become old at the same rate and to the same extent. Yeah, some people might have very sharp brain even until very old age or other way around. About 15% of the people over the age of 85 will get neurodegenerative diseases, but 85% of the people may not get any diseases, but they are still old. So aging happens so individualistically, right from the individual level to organ level to tissue level to cell level and within the cell to molecular level. There are no two molecules which become old at the same rate and to the same extent. No two cells become old at the same rate and to the same extent. Now, this is a fascinating observation, but which makes our field very challenging, because if somebody is looking for magic solutions, magic pills, one pill for everything all the time for everybody, well, that is a fantasy or that is a fraud, whatever you want to call it. So we know that aging is very heterogeneous and individualistic. And one beautiful way of saying it was done for the clinical data, for example, those of you who are aware of the clinical data, the scatter, if you have age and any measurement, the scatter increases with age. Older population has much wider scatter in any measurement to do. Whether some of the examples I have listed here where people have measured muscle mass, cognitive abilities, memory, stress tolerance, gait speed, the scatter increases with age. In younger ages, the populations or the individuals of a cohort are much more alike. They are not absolutely similar. There is a scatter, but this scatter increases. More we live, older we become, more different we become. And a Danish poet and philosopher said this statement in a beautiful statement. We are born as copy, but we die as original. Born as copy means that we are within the evolutionary restraints. There are not too many different ways of being born. But longer we live, more different we become. So from a human point of view, becoming old is a process of becoming authentic, original. And those are the social aspects. I will try not to go into those aspects here, which I often get very tempted towards. But we are born as copies, we die as original, basically telling in one sentence, thousands of papers' conclusions that aging is heterogeneous and individualistic. So what is then common? Are we so different? No, the common is our property, our ability to survive. We all use the word and we are familiar with the word homeostasis, maintaining the same state, which is actually a wrong term for biological systems. Biological systems are never static. We all know that. We all talk about that. But we use the wrong term, homeostasis, from machines, from engineering. It is actually homeodynamics. A child is born with certain basic requirements for survival. But there is a big danger zone where things can go wrong in early childhood. During growth, development, maturation, by the time we reach our reproductive maturity, this is what we have, a homeodynamic space, I call it, a green area. This is due to the property of homeodynamics. And the homeodynamic space, which is basically the three characteristics: stress tolerance, which is essential to our life. We are all the time under constant challenge from inside and outside. If our bodies can't take care of that, we are dead. Then damage management, damage control. The damage is occurring due to all the time the same processes. Oxygen, food, basic biochemistry creates so many errors, so many damage. If our bodies cannot have control over that, we cannot live. Then constant remodeling. This is a very important property for our immune system and many other systems, and even in the functioning of the brain. How do we adapt and change? Now, these are the characteristics which evolution has worked on according to the need of the essential lifespan, that they should not be affected, for example, in our species, in a significant manner before the age of 40, 45. Yeah? But then, when aging sets in, when the biological phenotype of aging sets in, what happens is, in the same way of representation, one can talk about aging is the shrinkage of the homeodynamic space, our ability to tolerate stress, our ability to control the damage, our ability to adapt and remodel is becoming lesser and lesser. And our red zone, the zone of vulnerability, that things can go wrong, increases. As a result, all those diseases, which in modern times in advanced societies are our main killers, or are the causes of all our suffering, are the diseases due to this process of aging. Whether it's neurodegenerative diseases, Alzheimer, Parkinson, or the bones, skeletal and bone-related diseases, osteoporosis, metabolic diseases, diabetes, arthritis, cataract, you name any major disease. The common cause of all these diseases is aging. We can definitely deal with one disease at a time. We will come to that. That's the biomedical approach to intervention. But if we really want to do something about our long life and healthy aging, we need to do something on the process of aging. So aging is the shrinkage of the homeodynamic space. That is the definition, at least I use in my research work. And why these things happen, why this shrinkage happens, why aging happens, there are again lots of theories have been in the field, and sometimes one can divide them into that there are central regulators, or there are specific organ-specific theories, or there are system-specific theories, or epigenetic regulation, or molecular pathways, the damage accumulation. But from mechanistic point of view, that why aging happens, what's the main mechanism and cause of aging, we generally agree that from aging point of view, it's the fourth one, the damage accumulation, which becomes important. I will come to that. What is important also is to, first of all, clear out that evolution has not made any genes to cause it. These things happen, but it is not because there are genes which are there in our bodies to cause it. Evolution does not select for gerontogenes. Evolution works on survival pathways, but it does not need survival pathways for infinity. So evolution works on what we call longevity assurance processes, which may look like gerontogenes because it's the same genes where things are going wrong. So this is another very, very important message from the biology of aging understanding, that there are no programs to cause aging, to kill the organism finally. Whatever those programs, cell death, apoptosis, etc., we know they are part and parcel of maintenance mechanisms. Apoptosis is very important during growth, development, maturation, during immunity responses. Yeah? But they are not the pathways, they are not the genes to kill me. They are the genes to keep me alive. The fact is, I will not be alive. I will be having problems in old age. So this is another very important achievement, that there is no single enemy sitting in the body which we can send a missile and become healthy and live forever. So this changes our viewpoint, how to deal with the research on aging. So the reason for shrinkage of the homeodynamic space and lack of any genes for aging is then explained by this fact, that imperfect maintenance and impair mechanisms. Evolution does not work on perfection. There is nothing perfect in evolution if you study scientific way of doing it. We are not perfect creatures. No other creature is perfect. Evolution does not need perfect systems. It just needs the system to do the function for that much, whatever the purpose is, whatever the essential lifespan is. That's why there is correlations between species with shorter lifespan and species with longer lifespan. If you compare any maintenance and repair system, there will be a correlation. The short-lived species have poorer maintenance systems than long-lived species, but there are no killer pathways, and that's very, very important. And this is what in the last 30 years, most of us have been measuring different types of damages, right? In the DNA level, whether these are mutations, epimutations, loss of telomeres, base modifications, whatever changes, those are under the change in the DNA. So DNA damage accumulates, very well studied area, and all the pathways. Same thing about RNA damage, protein damage and turnover, other macromolecular damage. That's a huge data. That is where all that metabolic pathways are coming in. And we understand which damage accumulates, which doesn't do much change. The problem with the damage accumulation so far is we don't know how much damage is tolerated by the system and how much damage is actually allowed to stay and how much can cause problems. Because again, from intervention point of view, we need to know how much to protect from damage. Yeah? If hydroxyguanosine is 5-fold increased with age, in whatever example you study, and I have a magic pill which can decrease, therefore, instead of increasing 5-fold, it is now only 2.5-fold. Does it mean I am half-aged? Have I become rejuvenated by half? No, that's what we will come to that. So aging research has achieved so much in understanding why aging happens, that's evolutionary reasons. How aging happens, these are the mechanistic aspects I have given you, damage accumulation. The third thing will be then, what do we want to do? And that's where the fact and fiction start giving us some problem. Are we still looking for the magical fountains of youth? Yeah, this is a 16th century German painting that there is a place, hypothetical imaginary place, where there is a fountain and where old people can come in, and at some stage they get fully rejuvenated and go about young. Very interesting social commentary here. In this picture, not even a single old man is entering into the fountain of youth, as if rejuvenation and healthy aging or healthy life is only women's problem. But that is what is society still doing it. A lot of things in the market are almost like these fountains of youth. Every day you will hear about miracle cures and this food or that drug or that chemical. Are we living in that fantasy? Or how do we want to proceed with this thing? So the question here basically comes from the same painting as a background. What do we really want to do? When we use the word anti-aging or aging intervention. And as a scientist, I would like to know from people and public and society, what do we really want to do with our age? Lifespan, natural lifespan is about 45. With our great medical achievements, we are able to now live almost double in most of the Western countries, and which the situation is changing in all countries. India is also having increase in lifespan all the time. But that's basically taking care of the early childhood diseases, infectious diseases, early deaths. So that things happen in 150 years where we have doubled up our expected lifespan than essential lifespan. Now, but if we live during that time, if I live beyond 45, aging phenotype comes in. I know it's sometimes strange to feel that aging, old age starts from 45. Yes, biologically it starts from 45. Sociologically, there can be very many ways of saying it. The early old age, middle age. But between 45, now I'm 65, I have been living in old age for the last 20 years. Although between 45 and 60, that's from social point of view, that's perhaps the most powerful and most enjoyable phase of modern human beings, where we have power, where we are the bosses, where we are doing all intellectual activity. In old times, even to reach 45 was a rarity. Average lifespans about 1,000 years ago were less than 20. Yeah. Very, very rarely anybody reached old ages. Now we all expect to reach old age. But what do we want to do when we are old or when we are having limited lifespan? Because, and I will die. So what do we want? Do we want to become young again? Is that our goal? That's where most of the anti-aging industry creates the fantasy of that rejuvenating and becoming young again. Is that what we want as society and as individual? We have to think about it. Or do we want to keep becoming old, but slowly, that the rate of aging should be slow, which is already happening due to better nutrition, better medical facilities. Now we say that the modern 70 years age is like the old 50. Yeah. So there is kind of a 20 years gain in health, but that's basic of social development, hygiene, medicines, technology. But the rate of aging, if we measure at any molecular level, has that been affected? We actually don't know. We don't know even how to do that. Then comes, we would like to become old, but stay healthy. Is that what we want? That all these diseases of old age, which are still a big problem, and socially that is from those people who worship money too much, then it's a big money problem, because it's the old age which uses most of the healthcare money. It's even discussed or analyzed that in our life, the amount of money which we spend on our health care, 90% of that money is spent in the last 1 year of life, and you still die. Yeah? If you have spent, say, 10 lakh rupees in whole life on your health care,
Not audible, sir. We're not audible.
Professor, check your mic. You are not audible.
Sir, we are unable to hear you, sir.
or later.
Suresh,
Only statistically. I know all the people you love, they are also going to die. We can't say when. It's a sad. The only compensation is all the people you hate, they will also die. So, shouldn't we get rid of the death completely? Is that science should do? Eliminate death through whatever medication. But we have to then think about it. How do we go about doing it? And that is where our approaches come. How do we view the biological system? Aging interventions and the body as a machine. Still, a lot of us believe in this old engineering metaphor, body as a machine, that there are different parts of the body, they work together, but if something goes wrong, you can set it and you can affect the whole system. And that is what in this drawing again comes, the previous drawing where we have described so many things, and now we think that on each target, we can either have stimulators or inhibitors or stabilizers or removers. If mTOR is the association, let me stop mTOR. If telomere is lost, let me put back telomeres. Or if this pathway is lost, let me activate that pathway. This is called the one-to-one mechanistic approach, a reductionistic approach. The problem is those target things don't go upward to the system. By increasing my telomere length, you cannot make me young again. By resetting some of the methylation patterns of some cell, you cannot rejuvenate me. But because the body has this complex interaction, adaptation, bypassing all these properties of homeodynamics. So body as a machine metaphor should be abundant. But unfortunately, most of us, including sometimes in my lab also, we take one target at a time, one chemical, one plant extract, one magic compound, and show the activity of this enzyme, that RNA going up or down, and then we say anti-aging. No, no, no. Don't kid yourself. That is not anti-aging. That is, yes, you are able to manipulate that pathway to some extent. So how do we go? How do we characterize aging interventions? Aging interventions can be seen basically the dominant biomedical approach, where is piecemeal remedies. What goes wrong, you try to set it. That is what science has done in the last 200 years, and it's a great achievement at whatever level, whether it's organ replacement or even modern stem cell therapies or even senescent cell removal. These are the piecemeal remedies that which can help when there is an acute situation. Most of the time, even stem cell coming from a very young, very lively stem cell population does not perform in the old body. So these are piecemeal remedies, which is a biomedical approach for treatment of diseases and management. Then there are, again, the most common one, which most of us are involved in, either replenishment. Whatever goes down with age, which hormone, which enzyme, which pathway, then I give something back to activate it or reactivate it and hope for the best. Most of the times, those things also don't seem to be working that much, except in the extreme, again, disease states, acute stage, but not as an aging process. Then the modern technologies are after all going to help a lot of people to have healthy aging by using better daily life equipment, mobility equipment. So ideally, what we need is this aging, which is the shrinkage of the homeodynamic space. Can we slow down the shrinkage, or can we regain some of the homeodynamic space again? And that's where you need holistic approaches, the total body approaches. One thing at a time. Target is very good for experimental purposes, for understanding the mechanisms, but they are not practical. More than 50 trials for getting rid of Alzheimer's plaque, beta amyloid, have failed, because the target was removing beta amyloid, and you remove it, person is still not healthy. And that's about even for a disease. And what to talk about aging and old age, which is much more complex. So we need certain methods which can work on the homeodynamic space as such. Not in homeostasis, homeodynamic. Things are changing, but then we can do something about it. And that's where I would like to introduce to the concept of this hormesis, which some of you may be knowing, some may not be knowing. Hormesis is the relationship between stress and health. We all know that high-level stress and continuous chronic stress is harmful and is the cause of many, many physical and mental diseases. But what we often forget is that the biology, our basic living processes, depend upon stress. Stress is the principle of life. Every time I breathe, I create stress, and that gives rise to a lot of complex biochemistry. And it is known that low level of stress, especially the stress of choice, if we are able to use, is good for health. We all know that. We all do that. We deliberately give stress to our bodies to gain benefits. You know, what is that? Physical exercise. Nobody doubts the grand advantages of physical exercise. I walk or I run and I get benefits at brain level. My cognition improves, my memory improves, my mood improves. How come? That's the holistic approach, that something happening even at one level gets amplified and affects it. This is called, in old terminology, you might be familiar with these kind of terms, biphasic dose response, U-shaped curves, inverted U-shape, adaptive response. In the textbooks, we are taught like there's no effect, but then linear curves or continuous toxicology curve. There are no curves which are of that shape in reality. Hormesis people, those people who work in the field of hormesis, have analyzed thousands of papers to show that everything has a biphasic dose response. The things which are toxic at certain levels are generally stimulatory or health beneficial at lower level. But please remember, it is not homeopathy. That is what it sounds like. But homeopathy works at 10 orders of magnitude below Avogadro number. So that's a totally different story. So we don't go into that. But hormesis is challenging the system. You give the stress, the body has all these hundreds of pathways of stress response details. And then if the stress is manageable, it becomes challenge, you get benefits. And this is the basic mechanisms behind hormesis. At the cellular level, there are basically 7 major pathways of stress response: antioxidant response, DNA repair response. So when anything challenges a system and the system tries to respond by one or more pathways, it can potentially be hormetic and it can be beneficial. We call all those conditions as hormetins. Any condition which by causing stress or mild damage can be health beneficial is called hormetin. Physical hormetin, exercise is the best example. Physical hormetin, you actually cause damage during exercise. You produce billions of more free radicals. You produce acids, you kill cells, and you get benefits which are whole body. So until 20 years ago, even people could not explain why exercise was good, because whatever you measured was actually bad. So in physical hormetins, exercise, heat, the sauna, even cold shock and radiation, these are the main categories. There is so much data showing that actually low-level repeated radiation exposure, just like low-level moderate exercise, repeatedly done, has beneficial effects both on aging parameters and longevity parameters. That's what radiation also does. Same thing, heat does it. Then from the brain point of view, which you are mostly involved in this conference, mental hormetins, a lot of things in the brain also can be beneficial. The stress in the brain can be beneficial. Yeah, whether you do brain exercise by playing chess, Sudoku, reading, or even meditation. There are some papers published, including the paper from one of the speakers in this conference, I think 2 lectures after me, Professor Gurchernikov, showing meditation having higher stress markers. The people who are in meditation, they actually have higher stress marker proteins like HSP. Same thing has been done in Hungary and other places, that people put into meditation go through the process of hormesis before you get benefits out of it. Very interesting topic, and you in IAN conference, I think you should consider that. What are the hormetic stresses? What are the hormetins which we can use to challenge our brain, to stimulate it, to make it more active and stay active for a longer period? Then there are lots of studies undergoing on nutritional hormetins. A lot of things which we eat in our food, they are actually good because they are bad. All spices come under that category. What is the nutritional value of even garlic, ginger, onion in terms of protein, carbohydrate, and fat? Nothing. Or this haldi. There is so much research on turmeric, curcumin. What is the nutritional value? 0. And what does it do? It increases a little bit of turmeric or the curcumin causes oxidative stress. It activates NRF2 pathways, then hundreds of antioxidative genes take over, and you get end result may be antioxidative, but the starting point is hormetic. All flavonoids, polyphenols, things like resveratrol, ginseng, ashwagandha, they induce low level of stress. This area is fantastically being developed both industrially and research-wise, and that is where there is fact and fantasy can be separated. Yeah? And the other side of the nutritional hormesis is not eating food, either low calories or fasting once or twice a week or intermittent fasting. What does that do? That activates autophagy. Autophagy is a stress response, and that is what gives you the benefits of that. Yeah. So this is one area where biological aging research is going in a big way, checking combinations of hormetins, 1 or 2 or 3 at a time, how to use it, what are the principles of hormesis. I don't have time for that. I'm already 37 minutes now. So I will be winding up telling the final message is that our aim in the field of aging is to have healthy aging, healthy old age, which will give us some advantage in the lifespan also in terms of number of years. But the most important part will be health span, that we do not have to go through one or more diseases for ourselves or for our loved one. Aim is maintain health, recover health, enhance health. To what extent, in what ways, and for how long? That's also the very important thing. Do you really want to live forever? Why not? If I can make you immortal, if I can get rid of death, wouldn't that be welcome intervention? So I wish you all the best for the next 3 days of fantastic conference of Indian Academy of Neurosciences, and please consider all these aspects in a holistic manner. I have just advertised some of my recent books, a popular book on age, which is also available on Amazon, some books on hormesis, and a very recent book explaining health. Because we talk about health, but we don't know what is health. Health is not just absence of disease. Health is much more than that. And what is that? Maybe you can discuss during this conference. ठीक है जी? Okay, shukriya. Thank you very much. Namaskar.
Thank you very much, Professor Suresh, for a very scintillating, exciting talk, which has excited our neurons, and particularly what you mentioned about the hermetins and hormesis. This is, I think, will give a lot of thought to the people and do it. So I'll now request Professor Raj Mehra to thank the speaker and any comments she wants to make at this time. Professor Raj Mehra.
Thank you, Dr. Seth. Thank you, Dr. Ratan, for a very thought-provoking lecture. You very elegantly covered the biology of aging, a topic of considerable interest to everyone today. When the average lifespan of humans has increased significantly, even in India, it stands at nearly 70 years for women. You have described about biological aging, social aging. The rate of aging is slowed due to social development, but at molecular level, it is a different story. I have a interesting reference here, better nutrition and aging. So there is a recent article in Science in relation to COVID-19 about a week ago, where people in India from 2 states, that is Tamil Nadu and Andhra Pradesh, people in India over 80 are spared of the disease COVID, which shows such people must have lived longer due to better nutrition and better immunity. Another thing, as you spoke, is we have hormesis challenging the system, makes hormones, mental meditation, mental hormisting, meditation, nutrition, hormones, etc. We may have some interesting questions in the chats. I will just open the chats now.
Ma'am, we don't have questions in this keynote lecture. Invited talks.
Okay, sir. Thank you very much. Thank you very much, Professor Suresh Ratan. It has been a wonderful lecture, and we all are stimulated to understand the aging process further. It is interesting to know, as such, there is no aging. It is all chemical changes happening at the molecular level. And thank you so much. Now, Anita, over to you.
Thank you, Professor Raj Mehra Madam, and thank you, Professor P.K. Seth Sir, for chairing the session. And thank you, Professor Suresh Ratan Sir, for enlightening us on the aging and how to go about further research through brain hormetins. we take short 10-minute break as Professor Ramchandran sir is setting up his slides. So he will be ready in 10 minutes. So, but be logged in and
I request if he's ready in 5 minutes, we start him at 5 minutes.
okay,
We have the time, very wonderful timing right now.
right.
As soon as Professor Ramchandran is ready, we
Ready.
should start the presentation,
Yes.
because there are a lot of people who want to hear them, and it's a Sunday evening over here. If
Yeah,
we delay it, we may lose some people. That
Sir,
is my request.
Sir, we help sir through a Zoom session in the night, but due to this pandemic, his assistant is having COVID positive, so he could not arrange his thing.
It's okay.
Sant is helping him arrange the things, and he will be coming on screen in just 5 to 10 minutes.
Dr. Anita,
Yes, sir.
I would request you to request Professor Suresh Ratan if you would be kind enough to write a review article on this.
Sure, sir. I will. Sure, sir.
Since we have 5 minutes, why
Yeah.
can't we introduce the chairperson?
Yes, that will be great, sir. Please. Even I was back of the mind thinking that that's a good idea. Sir, please, you can.
Kindly request him to do so. Yeah.
Professor T.R. Raju, sir, and Professor Pankaj Seth, sir, I request you to introduce each other.
Can I go first, Pankaj?
Yeah, you go first. You.
I'm truly excited to introduce my co-chair, Dr. Pankaj Seth, who is a professor at the Neurobiology Research Center. He is a very brilliant and gifted neuroscientist of India. And he is a fantastic friend of mine too, I must say. We are Twitter buddies too. So he has got research interests in the field of neuroimmunology, neurovirology, stem cells, neural stem cells, and glial biology. He is an expert to Department of Biotechnology and Indian Council of Medical Research. We work together in the ICMR expert committee meetings. The other thing common between us is we love to publish in Journal of Neuroimmunology. He is a board member and council member of International Society of Neurovirology, International Society of Neuroimmune Pharmacology, and Asia Pacific Society of Neurochemistry. I consider it to be a privilege to co-chair this session with Pankaj. Thank you, Pankaj.
Can I interrupt?
Yeah.
Pankaj sir, can I interrupt? Yeah. So actually Ramachandran sir is finding little difficulty. So he has requested the next lecture can go on, and then he'll be able to join. So,
I'm glad we don't have to give a lecture.
So, but we will have to check if Dr. Gulcharan Kaur is ready.
Okay. I will call.
I'll get an opportunity to reintroduce you, Pankaj.
No, I was saying you need to do that. I have to do my part, my honor. I will do it when Dr. Ramachandran is also there, so that you know. So is Dr. Anita, is Dr. Gurcharan?
Yes, Anita, I'm there. I'm ready.
Ready. Madam is ready. Gurcharan madam is ready. Thank you, madam. You always help.
Yeah, thank you. Thank you.
And what about Dr. M.K. Thakur?
Yes, we have to now contact the chairpersons.
I'm here. Please contact Dr. M.K.
Yes,
Thakur.
Shashi madam is there, and I will bring Thakur sir.
I think in the meantime, ma'am can start and while we connect to Dr. Thakur.
Yeah.
I'm trying to contact him now. हेलो आइए लाइन पे आइए डॉक्टर गुलचरण कॉल में हूँ उनको इंट्रोड्यूस करना है
So I think I can start.
Yeah.
आप शुरू करिए सर डॉक्टर ठाकुर आ रहे हैं
आ रहे हैं अच्छा
हाँ जी डॉक्टर लाइन पे आ जाइए हेलो
Good evening, Dr. Shashi.
Good evening, sir. How are you? Looking very bright.
I thought the lights are not powerful enough. Yeah, Sunday evening.
Dr. Raju is always glowing by the
Yes,
grace
yes.
of
Glowing in the dark only. Huh?
Madam,
Sir,
Can you-
you are here.
Yes, sir.
So, thank.
can you
Yes,
hear
sir.
me?
Yeah, sir.
Yes.
Dr. Thakur, you can start.
All right. So it's privileged. Dr. Anita, should we start?
Yeah, sir. First, you can initiate with introducing each other.
Yeah, we'll take over. It's my privilege to share this session with President Madam Professor Sasiwala Singh. She is the organizer as well as president of the academy. She is director of NIPER, and I think I don't need to introduce her further to the neuroscience community. So, madam, if you permit, we can go directly to the lecture.
I think I agree with Professor Thakur. We need not we know each other, and people also know. I hope. I think let's introduce the most important person for today's discussion for this distinguished lecture, Dr. Gurucharan Kaur. It is her time. It is her moment. Let us introduce her. So may I request Dr. Thakur to introduce her.
So, myself and Professor Sasiwala Singh welcome you all to this very prestigious lecture, B.K. Bachawat Lifetime Achievement Award lecture. This lecture is organized in memory of late Professor Bachawat, who was one of the stalwarts of neuroscience in India and former president of Academy. This award is instituted out of endowment funds created by members of Indian Academy of Neurosciences, and it started in year 2003. Since then, it is conferred every year to a neuroscientist based on his or her research contributions in the area of neuroscience. And to name few recipients of this award, previous awards, it started with Padma Bhushan Professor P.N. Tandon in 2003. And then we had Dr. B.N. Dhawan, Dr. Mehdi Hasan, Dr. P.K. Seth, and many eminent neuroscientists. Last year, it was conferred to Dr. K.P. Mona Kumar, and this year it has been conferred to Dr. Gurucharan Kaur. And so congratulations to Dr. Kaur. And Madam, would you like to introduce her or should I go ahead?
If you say, I can also,
Yes.
okay, I will introduce
Yes.
my dear friend, Professor Gurucharan Kaur. I know her almost from the year 1984 when we were staying together in the same hostel in All India Institute of Medical Sciences. It is a great honor and privilege for me to introduce her, such an exemplary personality. I think we'll know her very well, but in the interest of other audience who have joined us overseas, and I would like to say a few words about her. At the outset, congratulations, madam, for being conferred with a very prestigious award of Indian Academy of Neurosciences, B.K. Bachawat Award for the year 2020. I think 2019, right? If I am right.
It's 2020, right?
2020. Okay. She has been working in the area of neuroscience for almost 3 decades. She has immensely contributed towards it. She has been basically working about the natural products, their efficacy in protecting brain, and her papers are in very high impact factor journals. She is now is a university honorary professor in the Department of Biotechnology, Guru Nanak Dev University, Amritsar. Her interests are are basically benefit of various natural therapeutics agents in managing neurodegenerative diseases. I think she believes in coexisting with nature, and she is exploring the natural products, how they can be used for protecting behavior, especially the brain, in the disease condition, also when the disease occurs to the brain. This is what I think P.N. Tandon in his opening remarks said, which was a very, catching thing which he told us in his remarks today. She has been bestowed with a lot of awards and recognition. It's a long list. I will just name few of them. She is a UGC Young Scientist Career Award, which she got in 1993. She is Vice President of Indian Academy of Neurosciences from 2017 to 2019. She is a member of DST Expert Committee and many other committees of DBT, DST, ICMR, where she has been rendering her services as an expert. She is a Vice President of Society of Neuroscience, Neurochemistry, and co-chairperson Research Advisory Committee, Amritsar Medical College. Besides all this, I have known her. I know she is an outstanding mentor, researcher, teacher, and above all, a very nice human being. And we are privileged, and we are happy that she is also will be one of the person who will be in the category who have received B.K. Bachawat Memorial Award. With this, I want to give the floor to her. Madam, may I request you for your presentation?
Thank you, Professor Singh, for your kind words of introduction. And at the onset, I take this opportunity to convey my sincere thanks to the Indian Academy of Neurosciences, all the stalwarts, our very senior and founder members of the Academy, and the members of the academic council of the executive council of this Academy, which is very, very close to my heart. I have been associated with this Academy, and through this Academy, I have many friends in the area of neuroscience, which is the best part of being associated with this Academy. And I also take this opportunity to thank the organizers of this conference and wish them all the very best under these hard times of pandemic. They have been able to manage this meeting, and I'm sure next 4 days will be a feast for all of us. May I request Dr. Anita to kindly allow me to share my slides? Because I'm disabled, host has disabled participant screen sharing. Can you please help me?
Yeah, ma'am, we'll get you in very soon.
Okay. So actually, when I received this information, I received the mail that I have been honored with this very, very prestigious award, B.K. Bachawat Memorial Lifetime Achievement Award. And also looking at the title of this year's IAN meeting, from basic to clinical neuroscience. So I just scratched my head, what do I talk in oration for this prestigious lecture, as well as going with the mandate of this conference. So one thing which came to my mind was, let me talk about this very important key molecule, PSA-NCAM, which has been the subject of research in my lab for the last 20 years, and also where we have been able to show some results from the basic to the preclinical, where it seems like with little more efforts, these PSA work which we have done, I'll be sharing, can be translated. Okay, so the topic of my talk, as it goes, for the current award is to unmask the neurotherapeutic potential of this key molecule of neuronal plasticity, PSA and CAM. But before I start my talk, I must confess that all my whatever I have achieved in my life, the stage was set by my very honorable revered teachers, Professor H.K. Mangat. I joined the Department of Biology, Guru Nanak Dev University in 1975 for my master's program, and Professor Mangat was the first one to introduce me to the subject of neuroscience. Later, I joined JNU School of Life Sciences and was very fortunate to have Professor Bakur and Professor Rameshwar Singh as my mentors, supervisors for my PhD program. And after completing my, submitting my PhD in 1983, I joined Department of Physiology at All India Institute of Medical Sciences, and where I also was like introduced to many of the research scholars doing the PhD and postdoctoral trainings. And Dr. Shashi, as she mentioned in the introductory note, that we know each other since those times. And I was very fortunate to work under the mentorship of these great people. We all know that Professor Baldev Singh, known as the father of neurophysiology in India, and I must share with the youngsters listening to my talk that Professor Baldev Singh, at the age of 80, he used to be the first person to enter the department and the last person in the evening, late evening, to leave the department. And then, of course, I worked with Professor Chinna and Professor Mohan Kumar, and they introduced me to the subject of neurophysiology during my postdoctoral training. Until today, whatever I have been able to achieve in my academic pursuits, in my academic arena, these teachers have a great role to play. So whatever we have been doing in the lab and whatever we have been able to achieve in the lab is all the hard work. So whole soul credit goes to my PhD students. And till date, I have 24 students associated me with no gender bias, 50-50 males and girls and the boys. Okay, so with this, I now shift my focus to the work which we are carrying in our lab. So the main focus of our work is adult brain plasticity. So as you all must be aware that brain function is not about only the interplay of neurochemicals or neurotransmitters, but it is also the brain is undergoing structural changes, new synapse formation, exogenesis, cell migration. So all these properties of the brain, they are retained even in the adult brain, not only just during the neurodevelopmental stages. And that's why we call it adult brain plasticity. And
so
what
issue is not visible completely. The screen comes half cut. Yeah. Can you read just here now? Now it is coming.
Okay, I'll make it.
Okay, good.
Is it?
They are adjusting, I think.
Yeah, this is fine. This is very.
Yeah, this is better.
Okay, so in all this work which we have been doing in the lab for the last almost 3 decades, we have worked with this molecule under different physiological and pathological conditions to study its role, to know its role as a key plasticity regulator in the brain. So some of the areas which we have been working with are the what is the potential beneficial role of intermittent fasting dietary restriction regime, and some of the natural products. We have been working with 2 important Indian medicinal plants, Ashwagandha, Vidhania somnifera, and Tinospora cordifolia. And another line of action or research in our lab was to retask the FDA-approved small molecules and drugs as PSA mimetics, and we studied them for whether they are efficiently can help in recovery from the spinal cord injuries. Now, before I go to the core research, I would like to introduce this molecule for those who have not heard about or don't know much about this molecule. So PSA-NCAM basically is polysilylated form of neural cell adhesion molecule. So these polysialic acid residues, NCAM is a Ig superfamily protein, and on the 5th Ig-like domain of this NCAM protein, polysialic acid residues are added by the enzyme polysilyl transferase, and this addition of PSA molecules, the degree of polymerization varies from roughly 8 to 400. And that's why when you go for Western blot for this PSA-NCAM, you always find a smear, because the molecular weight will range from approximately 150 to 250. So because of the addition of these polysialic acid residues, this molecule as PSA-NCAM attains negative charge around, and also it has a high water binding capacity. So there is a high hydration volume around this molecule. So as a result of this post-translational modification of NCAM, addition of polysialic residues, it creates a sort of repulsive field around this NCAM, which otherwise is involved in hetero and homophilic interactions to stabilize cell-cell contacts and cell-substrate interactions. So this anti-adhesive property provides this efficiency to this molecule to participate or to be a regulator of cell migration, neurite outgrowth, and neurogenesis. So other than this anti-adhesive property, this molecule also plays important role in the regulation of ion transport, especially through AMPA and NMDA receptor. It controls the calcium channels, and all those downstream pathways are then regulated. And another important function it performs is because of the large hydration volume, this molecule as PSA-NCAM, it helps in retention and release of biologically active molecules like growth factors, the neurotrophins and neurotransmitters, and also closely interacts with the cell surface receptors like FGF receptors as neurotrophin receptors, and downstream pathways are then regulated through these interactions. So as a result of all these functions, this molecule is known to play a very, very important role in synaptic plasticity, in axon pathfinding, and one very important aspect is it's also regulating the neuron glial plasticity. In this area, we have been able to work quite a bit. So if I go into the go back to my work with PSA-NCAM, I was first introduced to this molecule during my short visit to McGill University Douglas Research Center under DBT Overseas Fellowship in the year 2000, where I took up a small project to study the role of PSA-NCAM in neuroendocrine plasticity. And we chose to work with the model of GnRH neurons. So you all know gonadotropin releasing hormone GnRH neurons, they are localized in the middle preoptic area of the hypothalamus, and the axon terminals from these neurons, they extend, in case of rats, almost 2 to 3 millimeters to reach the perivascular space, where they release the hormone around the vasculature, and then through this vasculature, the GnRH is carried to the pituitary, which in turn releases LH and FSH. And all this is under the control of peripheral steroid, sex steroid hormones by negative and positive feedback loops. So this is the background we started our work from to work on the hypothalamo-hypophyseal gonadotropin system. And during this time, one very important observation I made was that these, as you can see, this is the median eminence arcuate region of hypothalamus. This is the external zone and the internal zone. And these are these fibers you can see are the GnRH axon terminals. So during the proestrous phase and the diestrous phase, we compared the exogenesis, and clearly you can see here amazing results. The extension of these axon terminals, they extend, go near to the perivascular space. So this is the phase pro-estrus when the hormone is released into the circulation for taking this hormone to the pituitary for LH and FSH release. But during the diestrus phase, we could clearly see these fibers, axon terminals, they are withdrawn. So after making this observation, and during this period, I just want to share with the youngsters here that I was having great difficulty to find a good GnRH antibody clone. And out of 3 months, I spent 1.5 months just testing various clones. I was not successful. But then I came to know that in the same city, Dr. Robert Benoit, who had established a clone for GnRH antibody, LR1 antibody. So I approached Dr. Robert Benoit, and he was kind enough to give me a very small aliquot, which he could find because he had established this antibody in 1979. So after almost 20 years, when I contacted him, he gave me an aliquot, and to my surprise, this antibody worked for immunohisto at a dilution of 1:10,000. His recommendation was 1:50,000, but I tested 1:10,000. And with just 3 to 4 milligrams of antibody in hand, we could use this for over 10 years. So after this initial observation, what I did, I micro-dissected the median eminence arcuate region and the preoptic area, and we run the blots for PSA-NCAM, and we could clearly see there is downregulation of the PSA-NCAM expression compared between the pro-estrus to the diestrus phase. So we have the maximum expression during the pro-estrus phase. So to our pleasant surprise, we got this first paper in Brain Research on the neuroendocrine plasticity in GnRH release. So with this, we started our journey of working with the PSA-NCAM, and I got these 2 grants from time to time, 2002 and then 2007, one Indo-French and one DBT grant to work. So what we actually worked during that first 5 to 6 years was to understand how PSA-NCAM is involved in the remodeling of GnRH and the glial cells to facilitate the release of the GnRH into the perivascular space. So this is the external zone of the median eminence. This is the pro-estrus and the diestrus brain sections. So you can clearly see there is very high expression of PSA-NCAM, especially towards the external zone, which is downregulated during the diestrus phase. Just imagine an estrus cycle of 4 days, and in 48 hours, the expression is upregulated, again downregulated, again in the next cycle, the expression is upregulated. But what was interesting to see was that along with this upregulation of PSA-NCAM, there was also a lot of GnRH exon terminals, exogenesis, which were seen in the external zone, which was basically helping in facilitating the release of hormone. And then we also worked with how the glial cells, because our main aim was to look at how the glial and the GnRH exon terminals, they are reciprocating each other to help in the release of hormones. So what we observed was that the glial cell processes were withdrawn during the pro-estrus for GnRH exon terminals to get extended to the external zone of the median eminence, whereas during the diestrus phase, there were a lot of these glial processes seen in the external zone. And further, we studied how the expression of PSA-NCAM is being regulated by the neurotransmitters. So we studied the monoaminergic and GABAergic mediated expression. And here you can clearly see from the cell body, you can see very nice staining on the cell body of PSA-NCAM and the GnRH neuron. And then in the median eminence arcuate region, so we could clearly see downregulation of the PSA-NCAM as well as GnRH exon terminals in the median eminence region. And same was the counter-regulatory mechanism seen for glial processes. They were extended after the monoaminergic transmission was inhibited and the GABAergic transmission was promoted. So along with the expression studies for PSA-NCAM, we also went a step further to understand the mechanism behind. So we studied the transcriptional regulation of PSA-NCAM mediated neuron glial plasticity by studying the expression of this enzyme, which is the key enzyme polycellular transferase, which adds PSA residues to the NCAM. And there is cyclic changes. Again, we could clearly see the proestrous phase, upregulation of the enzyme, and diestrous phase, there is downregulation. Same is seen here in the northern blots. And further, we used another strategy wherein we use this endoneuraminidase enzyme, which cleaves PSA. So in the culture system, now GNV3 cells are conditionally immortalized GnRH cells. So we used a co-culture of these GNV and glial cells, and we treated these cultures, one set of cultures, with EndoN. And clearly, as you can see here, the PSA NCAM expression goes down. And with that, the remodeling of the GnRH neuron, GNV3 cell, which you can clearly see in this section of the slide, as it was downregulated. And again, Western blot clearly showed that there is downregulation of the PSA expression. And same was also confirmed by the in vivo studies, where we micro-injected endoneuraminidase enzyme. It was very, very tough experiment but one of my students, Sushil, was very, very efficient person, and he did this experiment, and really we got amazing results that injecting the endoneuraminidase in the median amines exactly abolished PSA NCAM expression, as well as it abolished reduced the GnRH exon terminals concentration or numbers in the external zone, whereas in the control group, there was very high expression. Now, having achieved this goal of understanding the role of PSA NCAM in neuroendocrine plasticity, we also went a step further. Another student, Sandeep, he started his studies working with the role of the expression of PSA NCAM. As we know, the PSA NCAM has prominent expression during the neurodevelopmental stages, and as we age, the expression of this molecule goes down, whereas NCAM expression remains global throughout. So in the adult, the PSA NCAM expression is restricted in certain regions of the brain, which show structural and functional plasticity. Like we studied the DG region of the hippocampus, median eminence arcuate region, and the piriform cortex. So you can clearly see from 1 month, 3, 6, and up to 24 months, there was a downregulation of the PSA NCAM expression, which is very clearly seen in the young animals. Same was true for median eminence and the piriform cortex regions. But on the short-term, late-onset intermittent fasting dietary restriction regime, we could see to some extent, as Dr. Kish has also mentioned in his talk, that these fasting is one of the hormetins which can revive or which can rejuvenate the system. So what we observed was there was, although very small, but yes, comparable to almost 18 months of age, there was upregulation of PSA NCAM expression. So this experiment with the aging rats and the rejuvenation with the dietary restriction regime we used really gave us edge to our further research on the role of PSA NCAM. So we went on further using the cell culture model system. Jaspreet Singh, for his PhD program, he took up this project to understand the transcriptional regulation of PSA NCAM, as I have shown earlier also, and you can see from this slide here, that PSA NCAM closely interacts with the cell surface receptors, AMPA and NMDA, and then regulates the calcium channels, flow of calcium, and further downstream signaling takes place. So it is not just the cell surface molecule, it is very much related or very much linked to the intracellular events. So he studied the role of NMDA receptor by using receptor blockers and low dose NMDA to show that yes, the PSA NCAM is transcriptionally regulated. So up till now, the observations which we made was that we could clearly see there are cyclical changes in the expression of PSA NCAM. So this is a molecule which is very, very important for neuroendocrine plasticity, which we observe across the reproductive cycles. And second was the molecule undergoes downregulation with age, but yes, it is retained in those brain regions which retain structural plasticity even in the adult brain. And you have seen the previous slide, even in the old age, the rejuvenation can be done. The expression of this molecule could be revived by intermittent fasting. And also we studied the transcriptional regulation by the NMDA receptor of this important molecule. Now, another very important piece of work which Hardeep did was that we started working with the natural products. And the very first observation which we made with the water extract from the leaf of ashwagandha, we call it ash wax, was that we Hardeep grew cystic glioma cells as a lawn of cells, and then he planned experiment to study the anti-migratory activity of this natural product from ashwagandha. So he created those confluent cultures and then gave a scratch, we call it wound scratch assay or wound scratch injury. So, and then followed by treatment with very low concentration of the water extract. And we looked at whether the water extract at very low concentration is able to inhibit the migration of these cells into the scratched area. So what we observed was, yes, 1% of the water extract, it was able to inhibit the entry or migration of cells into the scratched area. So here again, we took help from NCAM and PSA-NCAM because we were looking for whether these cells are undergoing neurodifferentiation, and no other markers are so good to tell us about neurodifferentiation than NCAM and PSA-NCAM, beautifully expressed on the surface. So we could clearly see, we used these slides where we had seen anti-migratory activity, same slides, cover slips we used for immunostaining, as well as we prepared cultures for Western blots, and we could clearly demonstrate, yes, these cells after treatment with the water extract of ashwagandha, they are undergoing neurodifferentiation. And as we all know, differentiation-based therapy for brain cancers is very, very important to bring the neurons back into their normal functioning. And same, we did work with the other plant, the active fraction from Tinospora cordifolia. And just I would mention here that this is the overlay for NCAM and PSA-NCAM, the explant cultures. They were treated with glutamate, which showed very clear neurodegeneration, but in the presence of butanol fraction of the Tinospora cordifolia, we got very nice neurite outgrowth and cell migration from these explants. And similarly, these glutamate challenged hippocampal neurons, they showed regeneration in the presence of butanol fraction of the Tinospora cordifolia. So this is in a nutshell the summary of what we had done till date, functional elucidation of PSA-NCAM. We tested under different physiological and pathological conditions using natural products, using regimes like IFDR, high-fat diet-induced obesity, brain cancers, glutamate-induced excitotoxicity, and we studied all the cell survival underlying pathways. And what we concluded from these results was that there is regulation of synaptic plasticity, axon growth, migration, remyelination being facilitated by PSA-NCAM. So having studied these various roles of PSA-NCAM, we were sure, and also there were a lot of reports in the literature that PSA-NCAM plays multiple roles. So at that time, we knew that yes, it is playing basically playing a multifaceted role in many of the conditions with cyclical changes. It is showing age-related downregulation, very high constitutive expression during the embryonic stages, and it is inducible because with the interventions, its expression could again be upregulated, and this is regulated both at the transcriptional as well as post-translational level on NCAM. So having looked at the role of PSA-NCAM in adult brain plasticity from multiple angles, next question which came to our mind was, this molecule is so important, it's playing very important role in various conditions. Can we exploit this molecule as a potential therapeutic candidate as well? That was our next question we asked. And it happened, like we all know that CNS injuries, the accidental injuries or sports injuries and otherwise, they are fatal because of the axonal retraction, neurodegeneration, and reactive gliosis leads to scar formation, which inhibits further healing or repair, and demyelination leads to poor nerve conduction, ultimately cell death and loss of function. And mostly these injuries are fatal. So this question when we asked, so I told my students to look into the literature, what is the status of PSA as a therapeutic candidate? And what we discovered in the literature was that there's a lab in University of Hamburg, Germany, Professor Malita Schachner. She is working with the mimetics of this molecule. Why? Because using the native PSA, which is derived from N. meningitis, which is the columinic acid, is the natural analog of PSA. So using the native form of PSA has its own set of problems in terms of immunogenicity. Also, PSA is susceptible to the action of endogenous neuraminidase enzymes. And also, if we go for synthesis, it's a multi-step, very tedious process. So what Professor Malita, I must share with you, she's can say a dawn in the field of cell addition molecules, professor in 3 countries, US, Germany, and China, and she has close to 1,000 research publications and several patents to her credit, working in the area of PSA-based therapeutics. So I was very fortunate that I would contact her at a time when she had just identified some of the PSA mimicking compounds. What they did was they, using ELISA-based screening and in silico approaches, they scanned some 446 small molecules. It says NIH Clinical Collection 1 library with the anti-PSA antibody 735 clone. They used the competitive ELISA with the columinic acid and identified some compounds. And she readily agreed to write a collaborative project. And as a common saying goes, where there is a will, there is a way. So it happened that during that time, there was a call for proposals between ICMR and BF in Germany. So we wrote this grant and we were very fortunate to get this grant also. And we started working with the small compounds which she had identified as PSA mimetics. PSA peptides as mimics are also have also been studied, but again we have problems with the peptides because of the size and their degradation. But these compounds are first of all, they are very well studied for many other, say, anti-neoplastic and migraine and all. So they are approved, FDA-approved drugs. So using competitive ELISA and in silico approaches, these molecules which were identified were 5-nanoxytryptamine, which is a selective 5-HT agonist, and then venorelbine and epirubicin, which are anti-neoplastic drugs. So having done this competitive ELISA rapid overlay to identify these compounds, the next task we set in was to evaluate the plasticity promoting potential of these molecules, small compounds, using primary neuronal and glial cell cultures. And then we used the in vivo paradigm of spinal cord compression injury in mice, whether these compounds are also effective to repair the locomotor dysfunction, loss of function in the mice. And later on, we continued this work with the help of a DST-SERP grant using the biomaterial approach. So we started this work using 2D and 3D culture systems, and the aim was to look at the right outgrowth potential of these test compounds. We started with 4 compounds, and ultimately we landed up with 5-nanoxytryptamine being the most potential out of these 4 compounds. So we used both 2D and 3D culture systems and showed the positive response. So I'll quickly go through these slides because we could clearly see they are causing defasciculation, they are causing new right outgrowth, and also they are interacting with the FGFR and neurotrophin receptors for the for facilitating the neuroregeneration and exogenesis. So as you can see clearly, using inhibitors for FGFR and pathway, which is a cell survival pathway, and also using NCAM knockout cell lines as well as NCAM, this PSA binding partner of Marx. It minimized the effects of PSA mimicking compounds on the neuronal cells. We used in vivo strategy also, where we used NCAM knockout mice. And we could clearly see the NCAM is essential for even for the potential beneficial effects of these small molecules. So having confirmed this, we went on to further testing the role of these small compounds in the wild type mice in the spinal cord injury model. So we could see the recovery in terms of locomotor functions of these animals using these open field tests and beam walk tests, as well as we did a lot of immunohistochemical and Western blot studies to find out what are the molecules which are involved. So as we all know, with the spinal cord injury, the major problems are demyelination of the axons and the astrocyte forming these scars, which inhibits the natural healing. So keeping those things in mind, we studied the potential for these compounds for reinnervation and downregulating the or inhibiting the scar formation by studying GFAP and IBA1 expression, and we studied reinnervation by studying monoaminergic, cholinergic, GABAergic, and glutamatergic neurotransmissions. Ultrastructural analysis for remyelination, and we did a lot of Western blot studies. So as you can see from this slide, it's important to note here that these animals showed almost 75% recovery, but the time it took was almost 8 weeks. So the time was very long, although recovery was there in the locomotor function. And along with the recovery, we also made sure that these caudal to the injury site, the spinal cord is undergoing reinnervation by the cholinergic, glutamatergic, and monoaminergic neurons. And we could clearly see these are TH positive neurons in the caudal injury site, and then the glutamatergic reinnervation and GABAergic innervation was downregulated, and there was very significant remyelination of the axons on ultrastructural studies. So having understood the role of mode of action of the 5-NOT and another molecule, venurelbine, using spinal cord injury and studying the downstream pathways, we could see there is cell survival tendency for these molecules to bring about remyelination and structural plasticity, and ultimately there was a recovery in the locomotor behavior. So having done this, our major concern now was that yes, these molecules are efficient in repairing the spinal cord injury, but the time it took was very long according to us. So we looked for some other approach, and we started using this biomaterial based approach using collagen and laminin. And what we did was that we used collagen and laminin gels, and the compounds were impregnated in those gels. Earlier, in the previous experiments, we were just flooding the injury site with the solution of the test compound and then suturing the site. But here we tested the same compounds, but impregnated in the collagen and laminin gels. So we made those hydrogels, and at the time of polymerization, the test compounds were added, and these are some of the benefits why collagen and laminin are specific biomaterials of choice. So these experiments in vivo validation of the neuroregenerative potential of 5-NOT was done in combination with collagen and laminin, and to our great surprise and to our we got this amazing output of these experiments. So instead of 8 weeks with the just flooding of the injury site with the test compound, compared to that, the collagen laminin impregnated compound, the recovery, locomotor dysfunction recovery happened in about 2 weeks' time. So we studied various behavioral or these locomotor functions to to ascertain our results, and also studied the reinnervation of the post-caudal injury site by studying the expression of NF200, which is a neurofilament important for which illustrates the reinnervation or exogenesis, and there was downregulation of the GFAP expression, or the glial cell astrogliosis was reduced. So again, this cell survival pathway and axon growth we could clearly see in these experiments. So insights from these work which we have done, it took us almost 20 years to reach this stage where we are now testing the efficiency of these small compounds as PSA mimetics. So insights from our research is that structure and function of PSA can be mimicked by small organic compounds, as they trigger the same signaling cascades as that of PSA, indicating that there is a possibility of retasking these molecules to treat nervous system injuries. And further, using the cross-linking chemistries with collagen and laminin as biomaterials and impregnated with the PSA mimicking compounds, they help in the controlled release of these compounds and also make sure the continuous delivery of these compounds to the injury site. So as a result, we got the functional recovery was speeded up. Now, before I end, I would like to say my pet words of healthy aging. I always like to talk about this area of research is also very close to my lab work and my own academic pursuits. So from all this, we can say, well, yes, brain retains plasticity even in the adult, but healthy lifestyle or healthy eating habits, physical activity, social integration these are the mantras for the brain to remain plastic and be rejuvenating. So let's move off the illness path and let's go down this self-management or self-management, self-help path for self-health, self-management interventions, so that we are able to sustain health and optimize our functional capabilities. So this is the reason I told in the beginning why I chose to talk on this topic of PSA and CAM. There were three major reasons. One is that we started working with this molecule in the year 2000. So it's 20 years. Exactly in September 2000, I came back from Canada and started work in my lab. So in September, when I got this information about the award, it was exactly 20 years. So this is one reason. And second is, I'm very happy to share that we published this review article in a very prestigious journal, Pharmacological Research, the neuroplasticity marker PSA and CAM insights into new therapeutic avenues for promoting neuroregeneration. And again, it was the month of September 2020, and I received this honor and opportunity to share my lab contribution on PSA research with all of you. And for that, I'm really, really very thankful to the Indian Academy of Neurosciences, all our senior members, members of the Executive Council, and the organizers of this conference, my dear friends Anita and Dr. Shashibala Singh. So this is Golden Temple at Amritsar, and all my dear students. And very happy to share that all my students who have already completed the PhD program, they are very well placed across the globe, and they are holding good academic positions as a faculty, as scientists, as people working with various other MNCs. So the grants from DBT, Indo-French, Indo-German, DBT, AST Japan, and DST SERB, and DBT fellowships are highly acknowledged. So with this, I again take you back to the entrance of my campus, beautiful campus, and very happy to share again. Last year, our campus was ranked second number in clean campuses across India. So you can very well see this very beautiful green campus and the spring in the campus. So with this, I close my talk, and thank you very, very much. Thank you.
Thank you, Dr. Hello, can you hear me?
Yes, yes, please.
Thank you, Dr. Gurucharan Kaur, for such a exhaustive and excellent presentation. And through your presentation, you have shown that PSA and NCAM role as a marker for brain plasticity.
Yes.
And the other point, which is really very excellent, is the way you have carried this research from bench to bedside. Like you are trying to understand this molecule, the pathways, then the way you have also entered into some of the mimetic molecules which can mimic PSA and CAM function, and how they can be used as a therapeutic. I wish you good luck, and I'm looking for a day when you have a product in your hand and say, this is what can be done, and this is how some of the senior citizens and people who are with the aging, if they can use your product, and we will be all proud of you. I'm sure you will do it one day. Thank you so much for a very excellent presentation, and again congratulations for being conferred with BK Bachawat Memorial Lifetime Achievement Award, I think, which you richly deserved. I'm sure everyone will be convinced after listening to her, the way she has carried her research from man to molecule, or from molecule to the man in the last 3 decades. Congratulations. I will also request Dr. Thakur if he would like to say something.
I also congratulate Dr. Kaur for delivering a very, very useful and informative lecture. And it was not only academic, but it was also a lesson for youngsters.
Yes.
How you have made the journey from the beginning to the end of this talk. And congratulations again, and all the best.
Thank you, sir. Thank you so much.
Thank you, sir. And over to Anita again.
Thank you very much. We had a really exciting talk. And
please unmute yourself. Please unmute, Anita.
so I congratulate once again Professor Gurcharan Madam, and we enjoyed your lecture, and another lecture by Professor Ramachandran is ready. He is online, and once again I thank Professor Gurcharan Madam for helping us by coming in the earlier slot. Thank you, ma'am.
Thank you so much.
So I will ask the chairpersons. No? So now over to Professor Pankaj Seth and Professor T.R. Raju sir.
Yes.
To take over for the Professor Ramachandran sir's lecture. They can introduce meanwhile. No?
Yeah, it's indeed a great pleasure to co-chair this session, this very important session where Professor V.S. Ramachandran would be speaking. I will just very briefly introduce my co-chair, Professor T.R. Raju, who's well-known.
Image froze.
Yeah, maybe he's trying to tell a lie.
And his major contributions has been in the area of ALS, especially sporadic ALS, where he has actually, his major contribution is of identifying CHIT1 as the biomarker for the sporadic ALS. With that, I would hand over to Professor T.R. Raju to please introduce the speaker.
Thank you so much for your kind words, Professor Pankaj Seth. It gives me immense pleasure to introduce Professor V.S. Ramachandran. He is unarguably one of the most outstanding neuroscientists of our generation. And currently, he is the director in Center for Brain and Cognition, University of San Diego, California. He has done tremendous amount of work in behavioral neurobiology, neurodegenerative disease, neurocognition, to name a few. He has been called later-day Marco Polo of neuroscience. That's a great honor. because he has journeyed through the Silk Road of science to strange, exotic cathays of mind. He is also called modern day Broca by Eric Kendall, none other than Eric Kendall. He is a fellow of the Royal College of Physicians, the Henry Dale Medal, which he got in 2005, and bestowed with Padma Bhushan by the President of India, and authored well-acclaimed one of the greatest books in neuroscience, Phantoms in the Brain. I am delighted to welcome Professor V.S. Ramachandran to deliver his talk on understanding the brain, challenges and excitement. Professor Ramachandran.
Thank you very much. I'm delighted and honored to be here. Thank you for the fine introduction.
Thank you.
And a little bit jet lag, having flown from California.
Oh my God.
So I'm going to talk about the human brain and its functions. And given the nature of the audience, I'm not going to go into the basics, basic aspects of brain function, but just concentrate on research that we've been doing in the last two decades at the University of California. And well, you think about the problem here. You've got a lump of jelly, the human brain, which you can hold in the palm of your hand, but it can contemplate the meaning of love, the meaning of infinity, the meaning of it, contemplating infinity, and so on and so forth. How is that possible? How does a miracle come about? Truly a miracle. How does it come about? Well, many approaches to studying the brain, including brain imaging, which is all the rave now. You see voyeuristic delight people take in looking at brain images in the newspapers when they come out. But there's one technique, and it's very useful. It's been very useful in the last 2 decades. Then there is visual neurophysiology, where you put electrodes in the brain, put an electrode in an awake animal doesn't feel any pain. You put the electrode, monitor the activity of individual nerve cells in the brain, and find out what the nerve cell is up to. What is it responding to in the world? It's called single unit neurophysiology. Whole visual system is mapped out this way by Hubel and Wiesel and Charlie Gross and others. Okay, then there is, of course, good old neurology. This is what I'm going to talk about. What I want to do is to take you on a journey back to the 19th century. All the experiments I'm going to talk about today in my lecture, for about 15 minutes or so, all the experiments could have been done by Aristotle. And certainly could have been done by Hewlett Jackson, Charcot, Freud. Any of these guys could have done it. Very low-tech experiments, but they didn't. Thank God, I get to do it. Okay. So what are these experiments and what's the goal? The goal is to understand the human mind. 100 billion nerve cells packed in the cranium, 100 billion nerve cells. Each nerve cell making 1,000 to 10,000 contacts with adjacent nerve cells. These points of contact are called synapses. They can be on, they can be off, inhibitory, excitatory. Based on this, somebody has figured out that the number of elementary particles, so the number of brain states, permutations, combinations of brain activity, exceeds the number of elementary particles in the known universe. This gives you some idea of the staggering complexity, the sheer complexity of the problem that you're encountering here. How do you deal with it? I told you about different techniques that are used. We primarily focus on neurology, believe it or not, just plain neurology, and sometimes with help from brain imaging, only when required. Trouble with high tech is it often, it's like you give somebody a new toy, some piece of equipment which hasn't been used before. What happens is, like a child, 8-year-old child, you give it a knife, go and cuts everything in the vicinity. There's no specific goal, right? You don't want that. When you're doing neurology, you do research in science, you want to be goal-oriented. You want to have questions, to be directed by questions, not by methodology, not by tools. The tools don't dictate the outcome. The questions do. It's a very important point, which is obvious to all of you, but I just mentioned it, okay? All right, so let's start with something quite simple. The Penfield map. Different sensory systems send signals to the brain to create maps of different sensory inputs, like touch map is somatosensory cortex, postcentral gyrus. You'll see a picture in the next slide, please. So parietal lobe, the brain has a parietal lobe. How do I go to the next slide? Thank you. Actually, I want the two slides below that. That's correct. Good. Okay, so you all know the structure of the brain. There's frontal lobes, parietal lobe, occipital lobe, temporal lobe. They're separated by big clefts called sulci or fissures. Now, it turns out that the somatosensory cortex, touch cortex, is in the parietal lobe, both left and right side, so 2 parietal lobes. And then the entire body surface is mapped onto the surface of the cortex. Systematic map is that. Okay, so that's been known for a long time. Now, what we found was essentially inspired by some early animal work. What we found was if you have a person lose an arm, so let's go to the next slide and look at the normal brain. Next slide, please. No, keep going. Keep going. Keep going. So, stop there. No, go to the next one. Next one. Yeah, good. Stop there. That's a coronal section of the brain. Coronal section, and you go through the postcentral gyrus, the big cleft on the side of the brain called the central sulcus, Rolandic sulcus, central sulcus. In front of the Rolandic sulcus, there is motor cortex, behind it is sensory cortex, including touch. You go there, there's a complete map of the body surface on the surface of the brain. How do you know this? You can take an awake human patient, like Penfield, the famous Canadian surgeon Penfield did this experiment. Awake human patients undergoing surgery for epilepsy, stimulate different parts of the brain, a tiny electrode, generating a very tiny current, often produces sensations. The person experiences tactile sensations. Oh, you're touching my elbow, doctor. You're touching my finger, doctor. You're touching my pinky, doctor. You're touching my palm, doctor. You're touching my face. You're touching my elbow. By stimulating different parts of the cortex, he obtained a complete map. It's a map you see that. It's called the Penfield map of the homunculus, or called homunculus, little man on the brain. Why is this important? Well, what we found was in this amputee, person with amputation, left arm was paralyzed for 10 years. 11th year, his surgeon decided to operate and they got rid of the arm. But the pain that was there in the original arm, the avulsed brachial plexus, persisted in the phantom with vengeance. In fact, increased after the amputation. So that doesn't serve any purpose, amputating an arm when you have pain in the arm, because the pain is in the brain, not in the arm. Okay? Anyway, this procedure was done, and he left with a phantom left arm, lying in a phantom sling, so to speak, but excruciatingly painful. So what's going on here? So I went and did the same mapping, touched different parts of his face and body, asked him, what do you feel? No equipment, nothing. He's just sitting in the chair, and I take a Q-tip, cotton bud, touch different parts of his face and body and ask him, what do you feel? What do you feel? My left forehead, doctor. My nose, doctor. My right chest. That's my left cheek. But wait a minute, do it again. That's my left index finger. That's my left pinky. That's my left middle finger. There's a complete map of the fingers, phantom fingers, on his face. Every point on the face, surface of the face that you touch near the jaw, lower part of the face, you touch, it evokes a sensation, precisely localized sensation, in the corresponding point in the phantom. So you have a map of the phantom limb on the face. Why would that be? This is a problem fit for Sherlock Holmes. That's what makes clinical neurology so fascinating. You got a problem here. Why would a person suddenly say that touching his face produces sensation in his hand, right? And when I looked at the Penfield map, the obvious answer was obvious. And indeed, some animal work had been done earlier by Mike Merzenich and Patrick Wall. What we found, what's going on, I think, is basically you amputated the arm. The sensory input from the hand does not go to the brain anymore. That region is devoid of. Is there any way I can point to a portion of the slide? Is there any way we can do that? Can I move this? I can't. This doesn't show. Does this show? Can people see that? No.
Yeah, cursor. We can see your cursor. Yes, Professor, go ahead.
So when the arm is removed, that region is vacant. No input to that region of the cortex. It's hungry for new sensory input. What happens is the skin from the face innervates the face area of the cortex. Normally, face skin, axons go to face area. But now they invade the hand area. It's vacated by amputation. You remove the hand area, so that's hungry for new sensory input. So this fibers destined for face skin invade the vacated territory originally occupied by the hand. So if you now touch the face, the message goes not only to the face skin, but also goes to the hand area and activates the hand area. So what happens? What does the subject feel? The answer is he distinctly feels the hand being stimulated. So you're touching my face, I feel it in my hand. Why is that, doctor? You have not noticed this before? No, I wasn't paying much attention to the where I was being touched and all that. It's very painful. That's the main thing that concerns me. Can you get rid of the pain? But let's wait and see whether that's possible. Looking at the diagram now is very interesting. So you remove the hand. I want to make sure you all understand that you remove the hand. That area of the cortex, the hand area, does not get any input. It's hungry for new sensory input. The skin of the face goes to the skin area, the face area of the brain normally. Now invades the vacated territory corresponding to the missing hand, activates those cells there. That sends a message higher up to the brain, but in consciousness you interpret it as being touched in the phantom hand. So that doesn't know that you remove the hand. Okay, so hand will be removed. It doesn't know you're touching the face. It senses its input is actually coming from the face. It still thinks it's coming from the hand. So whenever there's activation of the hand area, the brain signals that your hand is being touched. The fact that it's missing doesn't bother you. You just say the hand is being touched. Okay. All right. Now, so far so good. Now, next question is, what else can you do with this? If you now put cold water on his face, you take a Q-tip, dip it in ice cold water, put it on his face. You get again. You say, doctor, my thumb feels cold, icy cold. So the referral of sensations from the cortex, sorry, from the hand cortex, from the face cortex to hand cortex, referral of sensation from the face cortex to the hand cortex is modality specific, not higgledy-piggledy. The temperature, not only does the touch map, reorganize, but the temperature also gets reorganized in a similar manner. And lastly, you can take ice cube, you can take Q-tip in hot water, or you can take an ice cube, put the face, ice cube, different parts of the face. Oh my God, I feel that on my phantom thumb. That's on my phantom pinky, phantom index finger. Derek, do you also feel it on the face or only in the index finger? No, I feel it on the face as well, but the fact that you feel it in the index finger is strange and surprising. That's why I only tell you about that. But I feel dual sensation, feel here and in the index finger. So we already explained why this happens. The challenge is a dogma, previously held dogma for the last 100 years in neurology, that connections in the brain, human brain, are laid down in the fetus or in early infancy. And once these connections have been laid down, there's nothing you can do to change them in the adult brain. They're fixed. Why there's such little recovery of function and there is damage to the brain, the adult brain. Why neurology can be, until recently, was a gloomy subject. Not much you could do to help these patients. Because it was thought that no new pathways can emerge in the adult brain. But I'm here showing for the first time that the connections can emerge over a distance of several centimeters, 2 centimeters, the cortex. In the adult brain, in a matter of a couple of weeks, 2 to 3 weeks after amputation, you touch the face, the guy feels it in his missing hand. Over that distance, the guy has reorganized his brain. This means that the Penfield map is not some fixed construct. It's in a state of dynamic equilibrium with the environment, the skin, the sensory input. One additional point I'll make, so that there's modality specificity. Touch goes to touch, heat goes to heat, temperature to cold. Now my professor, Professor Pettigrew, very eminent scholar, who retired to Australia after he spent most of his life at Caltech in California, went to Australia, recently passed away, so I should have dedicated this lecture to him, but I will, next time I give a lecture, I will. Professor Pettigrew was a professor at Caltech, and he was very intrigued by all this. How did I get on to him? Oh yeah, he was sitting in Australia in a bar. My professor was sitting in an Australian bar. Then he saw a paper in Science, which I published, talking about this referral from the face to the hand when the arm is amputated, from face to the phantom hand. By the way, am I audible there? Am I speaking too fast, too slow? Should I slow down or should I shout more?
You are audible. Slowing down may help because sometimes there's a lag in the internet. So otherwise, everything is perfect.
Okay, I'll do it. Thank you. So what he saw this paper in Science sitting in a bar in Sydney with his friend, and he said, yeah, sorry. And what he said was, Ramachandran is saying that, you take a person with an amputation, you touch his face, he feels him in the phantom. Let's test it. So this guy was bewildered, the sailor sitting next to him, but he had a hook. He had an amputation and a hook. So Pettigrew had the sailor put the hook in a glass of whiskey with ice cubes floating on it. He said, "Oh my God, sir, my phantom feels ice cold." And then he put the ice on his face. He said, "My hook feels ice cold." Imagine there's a patient here with a stump and a hook. You now put an ice cube in front of his face and he feels the ice in the metal hook. It doesn't get any better than that in neurology, I can tell you. And we know the answer, why it happens. That's more important. Okay, now, so it's modality specific and all that. Now, what else can you show? You can also show, for example, that if you amputate the feet, look at the foot there. The foot is amputated right next to the genitals. And sure enough, when you amputate the foot, sensory input from the genitals, skin, cross innervates, invades the territory corresponding to the foot. So when you stimulate the genitals, it goes and activates the foot cortex. So the person thinks you're stimulating his feet and stimulate his genitals. And what's interesting is this step actually told me that he feels orgasm, sexual sensations, in his foot, in his phantom foot, when his genitals are stimulated. This nobody could explain until we came along and provided an explanation in terms of anatomy. Okay, and finally, if you sort of amputate the penis, which happens for all sorts of things, carcinoma, for example, or a jealous spouse. It's known to happen. I'm not making that up. Bobbitt case, Lorena Bobbitt. Then you get a phantom penis. If you then do a Babinski reflex or something, stroke his foot, he feels the sensation, stroking sensation in his phantom penis. So all these anecdotal observations are there, which make the study more interesting to the average medical student, but also have deep philosophical implications, I think. You'll recall Mriganka Su's experiments where he does the, well, I won't go into that. During discussion, we can go into Su's experiment. Okay, next slide, please. Okay, one thing I want to tell you is, no, go back to the previous slide. Go back to the previous slide. Sorry about this. Good, thank you. One of the things we noticed was the points, with this theory about what's going on, right? To create this reverse sensations in a patient's phantom limb. Important implication is new connections can be found in the adult brain, the adult human brain, with surprising rapidity. Question is, can you exploit this clinically to help patients with phantom limb pain? The answer is yes, and you'll see in a minute. But first, I want to draw your attention to something very curious here. Everything seems correct with this diagram, and my explanation seems sexy and seems on track, right? But always be careful to what your professors tell you. There's some loophole or some flaw. What is the flaw and loophole in my argument here? There is a loophole which I subsequently got past, but initially it was disturbing. Namely, you look at that diagram, Penfield diagram, where is the forehead? It's up near the hand, representing the hand. The peculiarity of the map. Genitals are below the foot, nobody knows why. The torso alone, the face alone occupies the biggest space in the entire torso, called cortical magnification. Mouth and lower face occupy a bigger area than the entire body, reflecting the greater utility of the hand and the mouth. Now, the exciting thing is, in the experiment, all our sensations, reverse sensations, came from the jaw. If you touch the jaw, upper jaw, lower maxilla, side of the face, that's when you see the sensation. You can see that in the next slide. Next slide, please. No, previous slide, previous slide. Can we go to the previous slide? Previous. The person, oh, that's very good. Thumb, index finger, pinky, you can actually map out the entire hand, missing hand on his face, draw the boundaries of the thumb, index finger, pinky, well delineated, clear cut, but like physiological receptor fields. You could be doing it with a cotton bud on a patient, but it's very, very similar to single unit recording. Very clearly delineated, and you put hot water, you'll say thumb feels hot. Now, notice it's all on the bottom side. But if you look at the diagram, and I said the reason is because invasion of from adjacent brain areas, from the skin of the face, invades the territory corresponding to the missing hand, activating the hand cell, so you think hand is being touched. Okay, but something wrong with it. Go to the next slide. The Penfield slide. No, sorry, the diagram. Maybe next one. Yeah, that's good. Do you notice the discrepancy? The head is upside down in Penfield's diagram. He mapped several dozen patients and he got this. So if I'm correct, the invasion from the adjacent hand area, so the hand area has been removed, sorry, hand input to the hand area has been removed. So invasion from the face territory comes from this side into the hand area, activates. But the forehead is what's near the hand, not the jaw. Where does it jump the forehead and go to the hand? You see what I'm getting at? If the face is really upside down, as Penfield claims it is, then all the maps we see in our patients should be in the lower jaw, should be in the forehead, not in the lower jaw. Clear? But always we see it in the lower jaw, not in the forehead. How do you explain this? We said two possibilities. Either Penfield is wrong or I'm wrong. Well, he took years to collect this data, so surely he must be right. But I said, no, I know I'm right, okay? This is vanity. So we then went and looked at the Penfield diagram. Each of those n dots represent one patient, one subject, one sample data point. So half a dozen data points he's drawn this diagram. Very little data, in other words. So this is a statistical fluke. So I said he's got it wrong. So a young man named Engel, Steve Engel in Stanford, was attending my grand rounds. He heard me say this. He said, I'm working on Penfield map again in normal humans. I'm going to check this out and contact you and be a co-author. So I don't want to be a co-author. You do what you want and tell me what you find. So he wrote to me about a week later. He said, Doctor, you're absolutely right. The map is upside down. Penfield's head is upside down. So now from this point of time, all the textbooks should be redone with his head going the other way. Now I'm very fond of this observation, not because it's a great theoretical interest. What does it matter if it's upside down or right side up? It's a quirk of anatomy. But it gives me sadistic thrill in spending 10 minutes on a patient and correcting the anatomy. That's what we've done here. That gives me a pleasure, maybe unwarranted, maybe. Now, can we have the next slide? Okay, before you go to the next slide, that's fine. Yeah, that's fine too. One of the things you see in these patients is a peculiar phenomenon, which we predicted from the literature. There's a group of neurons that have been identified in the primate brains of monkeys and more recently in people, called mirror neurons. Most of you have heard of this. Ordinary command neurons in the front of the brain, say the premotor cortex, originating in the premotor cortex, going to the motor cortex, sending signals down the brainstem, spinal cord. These command neurons are well known, discovered by Vernon Mountcastle a long time ago. But what Rizzolatti and his colleagues in Parma, Italy found was these command neurons, the monkeys grabbing the peanut neuron, pushing something neuron, pulling a rope neuron, different command neurons for different simple functions. The same neuron, a proportion of them, about 10% of these, will fire when the monkey watches another monkey performing that same action. So this monkey reaches, grabs a peanut. Peanut grabbing neuron fires. It orchestrates the sequence of muscle twitches that is required for precisely grabbing the peanut. Of course, regulated by cerebellar control as well. Otherwise, you get tremor. Now, what Rizzolatti found was 10% of these cells will fire when I'm not doing anything. I'm watching him reach for the peanut. My peanut grabbing neuron will fire when I watch him. It's called monkey see, monkey do neurons, or mirror neurons, subsequently found in humans. When I heard Rizzolatti give this talk, I jumped off my seat when I said, this is astonishing, most exciting thing somebody's discovered in the last 20 years. You're experiencing that monkey's feelings, or simulating it. You're doing a virtual reality simulation of the impending action of the monkey's hand in your brain, creating a simulation. What is the monkey up to? What's the guy up to? Mind reading, intentionality, all that is encompassed in the behavior of this neuron. Now, nobody's saying this one neuron is doing all that. It's a part of a network, but it's interesting that you can monitor at the level of a single neuron the properties of the network. And it's truly exciting discovery at that time. And I think, what can you predict from this? I'm always looking for predictions and clinical use, maybe clinical utility. But one of the things we noticed was that the mirror neuron, if you have a mirror neuron, I'm saying I have a mirror neuron. If somebody were to poke me with a needle, the neuron will fire. In thalamus, certainly in insular cortex, amygdala, various pain-related neurons will fire, or even touch. You just gently touch neurons in S1, S2. S1 is primary motor cortex, secondary motor cortex, S2, and finally superior parietal lobule. You don't have to remember these names. They're basically cascade of brain activity and brain structures in the somatosensory pathway. Now, what happens in this guy, as far as I'm concerned, when I watch him being touched, the same pathway is activated. So then, if that's true, why don't I feel it? If you feel it, if you watch me, if there's a gentleman sitting here, imagine there's a gentleman sitting here. If I touch his face, he feels it in his, or poke his face with a needle, he feels the poke activating his own brain, his thalamus, his insula, and all those areas. Anterior cingulate, neurons fire. And he says, ow! Now, what's the result is saying is if I watch him being poked, same pathway is activated in me. My mirror neuron fires. Even though it's my neuron, it's firing when he's being poked. That was breaking down barriers between me and him, the mainstay of much of Hindu philosophy. That there's no real fundamental distinction between your consciousness and my consciousness. What's dividing us is skin, skin and bones. So I like to call these Gandhi neurons. So what are these Gandhi neurons important for? Lots of things, but we don't have time to go into that. But the question is, why do I not feel pain when somebody is in pain? I experience empathy for the pain. I feel sad for him, but I don't say, ow, and withdraw my hand. I don't feel the pain that he's experiencing, even though my pain neurons are firing. Why is that? The reason is you've got normal skin vetoing the output of the mirror neuron, saying, look, you're not being poked, don't worry. Veto succeeds, you don't feel that person's pain in your hand, but you experience empathy. You inhibit the component, the empathy, the pain experience component without inhibiting the empathy. So you experience empathy for the person. I know what it feels. You're in terrible pain. I feel sorry, but you don't literally feel the pain. Okay, now, question. If you amputate my hand, then the inhibition is gone. The veto signal is gone. Therefore, I should now feel the pain in my phantom. In other words, if a phantom limb patient looks at a normal person being poked, he should feel it in his phantom. Extraordinary conclusion. Does it happen? Answer is yes. 3 patients I tested, left arm amputated, US Army, US Navy rather, phantom left arm. I asked him to simply watch me while I poke my hand, unexpectedly. And he's out. I said, what is I felt the pain, sir. Regarding the barrier between self and others using mirror neuron. Next slide. Next slide. Next slide, please. Now, if there is time, I'm going to come back to the phantom limb and I'm going to talk about some other interesting aspects of it. But given the time rush, I'm going to go straight to the second part of the talk, part 2. Part 1 emphasized plasticity, tremendous plasticity in the nervous system, over 2 centimeters change in anatomy. The books have to be rewritten. Now I'm going to talk about the stability of connections, the role of the genome in specifying connections in the brain. Next slide, please. Next slide. About 3 or 4 slides. Keep going. Keep going. That's just, yeah. I'm going to come back to this phenomenon of epitomophilia, where a normal person wants their arm amputated. Can you imagine that? A person, dean of a medical school came to me. He said, he has a secret he hasn't told anybody. Doctor, you might have some connections in Mexico. I want my left arm removed. And we explained this physiologically. And I'll come back to it later during discussions if people want to hear it. Next slide. Keep going. Next slide. Next slide. Next slide. Okay, pause here for a second. I'm not going to go into this other widely publicized old work about mirror therapy for phantom limb pain. Diane here has also collaborated on the original studies. So patient has a phantom limb. Often he says, my phantom's frozen, doctor. It refuses to buzz. It's painful. It's in an awkward position like this. He shows me the normal hand. It's like this, my phantom. And a normal hand cannot adopt that position, an anatomically impossible position. But the phantom is adopting the position. Then I put a mirror on the table, put the normal hand on the right side of the mirror, so you see the reflection of the normal hand where the phantom used to be. So the phantom is brought to life using a mirror. You see the mirror reflection of the phantom, the real hand, optically superposed on the phantom. Now, if you move your real hand, send commands, bilateral commands, you're going to get a visual impression that the phantom is moving for the first time in the last 5 years or 10 years, or 1 year or 6 months. But not only does it look like it's moving, it starts feeling like it's moving, and the pain gets relieved very often, immediately, or sometimes after a few weeks, in about half the patients. That's surprising enough, and you can go and read the literature on that. There have been 80 clinical trials on this, about 40 of which are good trials, and the procedure works. Now, another condition, RSD, which most people don't know about, but an intact arm with a tiny injury or a stroke affecting the pathway, somatosensory pathway from the arm to the brain is affected, producing excruciating pain in that arm after stroke or after injury to the arm. 2 kinds of RSD. And then normally the injury heals. There's inflammation, there's swelling, there's redness, there's warmth, all the signs of inflammation. To immobilize the hand reflex immobilization of the hand. So you can't move it. You don't want to move your injured hand to worsen the problem. It's nature's way of temporary paralysis, functional paralysis of the dystrophic hand. But not only that in some people, most people recover. Inflammation subsides, goes away, fracture. It's a metacarpal bone fracture, tiny fracture. Fracture, the inflammation, all the signs of inflammation, warmth, swelling, pain, immobilization, all that. For a week or 2, 3 weeks, then reverses, bone heals, everything goes away. But in 1% of patients, bone does not heal. The inflammation remains. Fingers start swelling up. Hands start swelling up. Entire arm swells up, excruciatingly painful, completely paralyzed for life. And there have been dozens of treatments tried, invasive, spinal cord transection, thalamotomy, not rhizotomy, dorsal rhizotomy, all these procedures have been done, usually to no avail, no compared to placebos. You put a mirror there, and we've suggested this a long time ago, and it's actually been implemented by Patrick Wall, the late Patrick Wall, and others in Australia, in Australia. What they found, in Oxford especially, Peter Halligan, what they found was if you put the mirror and you put a dystrophic hand, a real hand, painful, intact, painful hand, look at the mirror reflection, move your normal hand, it looks like the paralyzed hand is coming to life, it's moving. It's not actually moving, of course, because it's paralyzed, but you create the optical illusion that it's moving. What happens? It actually starts moving. So much of the functional paralysis is functional. The visual feedback kicks in, and the arm starts moving online. As you watch on the table, it starts moving. The pain, surprisingly, is reduced because part of the pain is the discrepancy between the command and the arm refusing to move. Move, no. It's stamped into circuitry of the brain. This discrepancy is removed by putting a mirror there. You move your normal hand, you close the loop, it looks like the dystrophic hand is moving with impunity, and soon you find that the swelling subsides. That's very important. The swelling, the physical signs of inflammation subside. You can't fake that. You can't have placebo swelling. So this rules out any possibility of placebos being involved as the primary factor in reduction of pain and RSD and swelling and inflammation. Okay, that finishes all the clinical stuff. Now I want to talk about extraordinary phenomenon seen in normal people, 1 out of 50 normal people, more common than previously believed. People thought it was 1 in 2,000, 1 in 1,000. Actually, it's seen in 1 in 50 people. Called synesthesia, discovered by Francis Galton's first cousin, Charles Darwin's first cousin, Francis Galton, who founded eugenics. Slightly nutty guy, but brilliant nonetheless. Okay, what did Charles Darwin show? I mean, what did Galton show? Certain percentage of normal population, otherwise completely normal, could be your neighbor, nothing physically wrong with him, but one peculiar quirk. Every time he sees a number, he sees a color. 5 is red, 6 is indigo, yellow is chartreuse. 9 is chartreuse. 6 is something else, brown. Each number is tinged with a particular color. It stays constant throughout his life. Completely harmless, except when he's trying to remember phone numbers, it gets jumbled. Sometimes it helps them remember phone numbers. When you ask them to remember a phone number, they see in front of them colored rainbow-like pattern. They rattle off the colors and they remember the phone number, unlike the way you and I do it, okay? It's very compelling to this guy, but people said they're crazy. Synesthetes are crazy, they got schizophrenia. Many synesthetes were misdiagnosed having schizophrenia, when in fact it's a specific condition called synesthesia, as Galton pointed out. Now, why does this happen? Why would somebody say that? No. A couple of things I want to warn you about here. In the neurology clinic, psychiatry clinic, you run into all kinds of fascinating cases. Some of them are worth pursuing, turn out to be gold mine, like Capgras delusion, which I told you about, which I haven't told you about, lack of time, or apotomophilia, which I haven't told you about. You invite me again and I'll tell you. But there is another condition called, another example is synesthesia. Right? But for every real syndrome, there are half a dozen bogus syndromes. Whether there's malingering or not even malingering, unconsciously playing up bizarre symptoms. And a good example, I'll give you 3 examples of bogus syndrome. You have to be very careful in neurology not to spend your life, waste your time studying these syndromes. So how do you know that synesthesia is not one of those? Well, I'll explain why in a minute. But let me give you a clear example of bogus syndrome, right? Well, okay, de Clerambault syndrome. De Clerambault syndrome. What is de Clerambault syndrome? Anybody here know in the audience? I guess you can't respond here. It's not a regular hall. De Clerambault syndrome refers to a young woman who develops a delusion, a fixation, that a rich, famous man is madly in love with her, but is in denial about it. I'm not making this up, the actual diagnosis. Okay. Now, I can assure you there's no such syndrome. Because people do behave like that, but it doesn't mean it's a syndrome. And ironically, there is no converse of the syndrome, where an older guy at the end of his life develops a delusion that this young hottie is madly in love with him, but is in denial about it. That's much more common, even among my colleagues, but it doesn't have a name attached to it. You see where I'm heading with that, okay? Now, the second syndrome, which I think is almost comical, chronic underachievement syndrome. What does that mean? Stupidity. In my day, it should be called stupidity. There's no chronic underachievement syndrome. Take it from me. Third syndrome I'll tell you about is the best one of all. It's called oppositional defiant disorder, ODD. Now, what this means is a spirited youngster dares to challenge an authority figure, like a parent, a father, or indeed the professor, or his parent, or his own father. He dares to challenge it, or his professor, or his physician, his psychiatrist, an authority figure. The man who invented this syndrome is a genius. Because any attempt by the patient to protest the diagnosis can be construed as additional evidence for the diagnosis, right? So it's a no-win situation. And neurology and psychiatry are full of examples of this kind. You have to be wary of. Okay, now, what causes synesthesia? Let's go back to synesthesia. What causes it? Well, first of all, as I said, synesthesia is seen 1% to 2% of people. Numbers are seen as colored. And we found that this is real phenomenon. The way we showed it was real was, next slide, please. Show first, so every phenomenon we discover, every syndrome we discover, first, is it real or not? Is the patient making it up? Very often, it turns out you're not smart enough to figure it out. The patient is not making it up. So don't jump to the conclusion the patient is making it up because you don't understand what's going on, okay? Number one. Number two, how can you help him? If it's troubling him, can you devise a clinical procedure that helps the patient, like phantom limb pain? Third, what is the underlying neural mechanism? You establish this phenomenon is real. What's causing it in the brain, the symptoms? And fourth, very important, what are the general implications of this? Is there any overarching message in this syndrome that I've been studying? Or am I just studying it for its own sake? These four questions loom large every time you study a patient in neurology or psychiatry. Okay, this is about phantom limb pain, quantitative study. I'm not going to go into. We're going to look at Tsao's paper, TSAO, Tsao, Walter Reed. Next slide, please. Reflexive RSD, inflamed, painful hand, starting with a tiny little fracture, becoming permanently like that. Look at the size of the hand. Next slide. Another example of RSD. Next slide. By the way, there's lots of clinical trials showing this works beautifully. I wish people had adopted it in India. It requires a mirror. That's all it requires, just a mirror. And some training by the OT, training the patient. Next slide. Next slide. Good. That's RSD. Okay. Mirror feedback can be used for RSD, stroke, even for stroke, hemiparesis, painful strokes, Disagin-Gusto syndrome, post-stroke pain, or just pain in a peripheral nerve injury pain. All of it has been treated with mirrors. Some of it's going to be true, some of it will be false. I don't know which is going to hold up. But RSD has held up clinical trials, controls everything. Next slide. Go back to, okay, keep going. Keep going. I already told you about when you remove the phantom hand, when you remove the arm, you have phantom hand, you watch somebody else touch their normal hand, you feel that sensation in your hand because your veto signal is gone. The hand has been removed, you're a phantom. You watch somebody else being touched in a real hand, your phantom experiences that sensation. And conversely, if you've got itching in your phantom or pain in your phantom, you watch another person being massaged, you feel the phantom massage, and that reduces the pain, sometimes eliminates it. We're testing all this in the clinic right now. Keep going. Good, synesthesia. Stop there for a second. Okay, keep going. Good. How do you prove this syndrome phenomenon? We found 2 students in our class who had 2. The number 2 was red, and 5 was green. I said, "What do you mean 2 is red? What exactly do you mean by that? You can see it. Are you sure it's red? No, it's black and white. But I see it in my mind's eye, and I see it tinged with the color. I can only see the color around the letter, but I know it's actually black. It's like a transparent sheet of red is put on it, something like that. I see dual color, red and black, similarly red and green, red and black and red, black and green. Okay, how do you prove he's not making it up? You showed him this slide. Made up of a bunch of 5s. Amidst the 5s, there are a bunch of 2s dangling in there. Can you spot the 2s? It takes a while to see the 2s. Then I ask you, what do the 2s form? Do they form a shape? The answer is yes, but you can't see it. Normals like you and I can't see it. But you give it a synesthete who sees 5 as red and 2 as green or whatever. Next slide. Next slide, please. He sees immediately sees a red triangle. I haven't done anything to the picture, but if I'm a synesthete looking at it, 2 are reds and 5 is green. Instantly he sees that inverted triangle or square, whatever you show him. It pops out from the background because of the color induced by the synesthesia. No normal person can do that. Therefore, QED. The guy is not making it up. He's got real colors being created out of the numbers. Why does it happen? Okay, next slide. Why does it happen? Well, we looked at the brain. It turns out you go into brain anatomy, you go into fusiform gyrus in the temporal lobes. That's where numbers are stored. Visual appearance of numbers is stored in the number area, shown by Stanislaus Dehaene and Tim Rickard in our center at UCSD. So the red circle there in the fusiform gyrus represents the number area of the brain. Green patch of Semir Zeki involved in color processing, color area. Why are they right next to each other? What's the likelihood? The most common type of synesthesia is number color synesthesia, and those 2 areas right next to each other in the brain. Huge brain, right next to each other, almost touching. This is very small chance of coincidence. This is a real explanation. In the fetus, early fetal life and early childhood, every part of the brain is connected to everything else. Tremendous excessive connections, redundant connections. These are pruned away by pruning genes, excess connections. So you remove the redundancy and you create the modularity characteristic of the normal adult brain. Is that clear? You sculpt out all the junk, create the normal brain. Then you end up with the fusiform gyrus, color area, number area. Now, if the pruning gene gets mutated, or the mutation is selectively expressed in specific regions of the brain, not throughout the brain, some parts of the brain will have defective connections. They'll have no connections, the pruning or excess connections. Pruning has not occurred because of mutation. Therefore, the number area, if it happens to be expressed in the fusiform gyrus, number color synesthesia. Every time you see a number, it's going to cross-activate the redundant connections, going to cross-activate the color area. Every time you see a number, you see a color. Is that clear? Then you can do brain imaging. You can do brain imaging, and you take the patient, and then you put him in an MEG machine or an FMR, and then show him black and white numbers. There's no activity. There's only activity in the number area, the red circle. Show him color, and then you get activity in color area and number area. But in the synesthete, you just give him black and white letters. The color area also lights up, clinching the diagnosis. I mentioned that imaging studies are notoriously prone to error, human error as well as machine error. So we do it only when it's absolutely essential. But there's a mania concerning imaging which you have to be wary of. Anytime there's a mania in science, you have to be careful. Okay, now next is, you've shown this. Let's go a little bit further. There's a nice slide. That slide is showing some very important principle. Yeah, okay, what's going on here? This shows cross-activation between number and color area. Number area is green, color area is red. Cross-activated in synesthetes alone. Next slide. No, go backwards, sorry. You're almost done in a few minutes. Go backwards. Backwards. Yes. So numbers and letters of the alphabet evoke colors, okay? Now, what I found was a dean of a medical school came and said me, and he said, I see colors with numbers. I don't know what it is. People said you diagnose very rare things. And I said, yeah, I can tell you what it is and explain it. But he said, but doctor, you're not explaining to me why I see colors only when I see numbers. And I see strange colors which I don't see in the real world. Colors only seen when I see numbers. The reason is the number grapheme, black and white number, bypasses retina and all that, goes straight to the color area in the brain and hits the color area. Because there's nothing wrong with the receptors in the brain. Colorblind guy has receptors missing in his eye, deficient pigments, therefore he's colorblind. So when he looks at the world, he doesn't see all the colors. He sees a very pale imitation of the world. But the numbers go and activate the color area in the brain directly, producing colors which he cannot see in the real world, but only when he sees numbers. Direct proof of my idea of cross-activation. Now, another example here, which you experience for yourself. Now, how many of you see what that is? Can you read it? How many of you can read it? Raise your hand. I can't tell you raising your hand or not, but just for fun, raise your hand. Okay, that's 90% of you. Now, if you don't read it, then squint your eyes. Squint your eyes and make it blurred, nicely blurred. Almost barely visible. Now read it. No sex causes bad eyes. How many of you see that now? Now, the interesting thing is, you show it to a synesthete, very quickly he says, oh, I see colors here. I don't see any letters, but I see colors, red, green, blue, yellow, pink, yellow, blue. Oh, it says no sex. He sees the colors before he sees the numbers, the shapes, which means the cross activation in the fusiform occurs prior to conscious awareness of the number grapheme. Next slide. We're almost done here. Next slide. Keep going. Keep going. Stop there. Okay, so why is synesthesia important? Who cares? Some people saying colored numbers. It's important because incidence of synesthesia. Synesthesia is 8 times more common among artists, poets, and novelists than in the general population. Why? What do artists, novelists, and poets have in common that all of us don't have? Many of us don't have. They're good at metaphor, analogy. Linking two seemingly unrelated ideas is a metaphor. It is the yeast and Juliet is the sun. Juliet is not the sun, and she's not a burning ball of fire. She's radiant like the sun, warm like the sun, nurturing like the sun, rising in the east, rising in bed like the sun rises in the east. Your brain can form all sorts of connections, and Shakespeare was a master at metaphor. Hence the evocative power of Juliet is the sun. Now, what I'm claiming is if the cross connection between different brain regions occurs locally because of transcription factors in the fusiform data, you get color, number, synesthesia. Every time you see a number, you see color. But who cares about that? If it's expressed diffusely, you can get a greater propensity to link seemingly unrelated, far-fetched, far-flung regions of the brain, representing different ideas, sensory qualities, concepts. There's a greater opportunity for linking these ideas. And that's what you mean by creativity or metaphor. Metaphor is the ability to link two seemingly unrelated ideas. Therefore, these patients, I don't know, not patients, these individuals are more prone, more effective at generating metaphors than the rest of the population. Hence, the 8 times higher incidence of artists, poets, and novelists of synesthesia in these people, QED. So now you can clone the gene. People have started cloning the gene. What's exciting about this, you can start with one quirk in the brain. Somebody has a quirk, saying numbers are colored. You think it's not important. You start studying, show it's real. After showing it's real, you say, what is the mechanism? Fusiform data, cross-wiring, mutated gene, defective pruning. And then you explain Shakespeare, creativity, metaphor, taking you all the way from the gene, identified large in the population, all the way to Shakespeare metaphor creativity in one preparation using Q-tips and colored pieces of paper. All right. That another phenomenon closely related to this, and then we're done. This is called a calendar lamp. So synesthesia occurs not just between number and color, also occurs between number and position, or days of the week are colored. Sunday is blue, Monday is yellow, Tuesday is chartreuse, Wednesday is indigo. The color persists throughout your life. People say it's because originally people who are studying this, my colleagues, they say numbers are colored because they play with refrigerator magnets in childhood. You know, magnets are colored magnets you put in the refrigerator. And I said, well, if that's true, why don't we all have synesthesia? We all look at magnets, play with magnets. Number one. Number two, why does synesthesia run in families? So the same magnets are being passed down? Doesn't make any sense. Common sense is very important in science, believe it or not. Anyway, it turns out that that's not the explanation. It's not colored magnets. Some people said, Rama, don't waste your time. They're all junkies. They're mad, saying 5 is red. Golden rule, if somebody tells you something peculiar like that in medicine, psychiatry, and neurology, and you don't understand it, don't think he's mad. Usually it means you're not smart enough to figure it out. Probe deeper and you'll find out what's causing it. It can lead you to a gold mine. Here's an example. Number calendar synesthesia. Every month in the calendar occupies a location in space. If I ask you to imagine the annual calendar, anybody in the audience, annual calendar. Imagine an annual calendar. Tell me what it looks like. Most of you will say it's a rectangle, grid of rectangle, January 1st, December 31st. Okay? Going left, right, for 4 weeks within each month. So you got a monthly calendar, 12 months. And then it's laid out in hula hoop. You had a rectangle, blurred, barely visible rectangle in front of you. You don't actually see it, you just imagine it. These people said, no, doctor, I actually see it. I don't imagine it. I know the difference. If I imagine a cat, I can imagine a cat. Here I see the calendar in front of me. Crisply defined outlines, black thin lines. August, September, October, November, December, January, February. Clearly printed there in the sky that I'm gazing on this visual calendar in black and white, high contrast, in a specific font, Helvetica font, Arial font, specific font, the letters are for all the months. What's going on here? No wonder people thought they were crazy. What do you mean I'm seeing fonts in front of me? But they're not crazy. I'll tell you why. How do you prove he's actually seeing that? Maybe he's making it up. How do you prove it? But people have been arguing about this for about 100 years, since Galton's time, but this is just bullshit. There's no real phenomenon here. Well, what we said was we just ask him to think about the year, the hula hoop, or something L-shaped, like a Lego, L-shape, test tube shape, different shapes of calendars. Why should that be? So this lady had a circle one, another lady had a L shape. I just want you to name the months of the year quickly, starting from August, September, October, November, December, January, February. They do it. Obviously, they can do it, like us. Now comes the bit. I want you to name them backwards, skipping one at a time. So October, August, June. You get it? Going backwards, skipping one at a time. You and I take minutes. Very hard to do. The sequentiality goes backwards. It's very hard to do. They do it at the same time, do it faster. Why? Because they're seeing it actually in front of them. They just have to read it out. They don't have to do an algorithmic computation in the brain. Spatially, it's laid out from, they read it backwards, it's just as easy as reading forward. This clinched it and showed that, in fact, these people really are seeing this pattern. Why should they be seeing this pattern? God alone knows. But what are the other implications? Well, I'll show you a couple of other slides and we're done. Next slide. Very important implications. Next slide, please. Please. Next slide. First of all, I should mention that Sienna Colson, Coxside Department, and Ursina Teuscher, Zeev, and I'm leaving out of it, Tony and Brang, David Brang, all of these people were involved in the study. Okay, so what's going on here? I think that the calendar is represented somewhere in the brain, and I'll tell you where it's represented in a minute. I showed you it's a real phenomenon. The question is, where is it? What do you need it for? Well, obviously, you need it for daily use, your calendar. Put yourself in the appropriate context in space and time. Now, the structures involved, I claim, are the hippocampus. Hippocampus, we think only as memory. In fact, it's very much involved in place and time, which is involved in memory, of course. Anything you encounter in memory has a location, label, an address in time and space. Shades of relativity here, but I'm sure it's a coincidence. Okay? So, okay, so what? Hippocampus is also involved in sequentiality. And now they found cells in the hippocampal grid cells and the play cells, which is an awake rat wandering around the cage enclosure. You put electrodes in the brain, monitors the activity of play cells. When the rat goes to a particular place in the cage, the cell will fire. No matter which direction, where he comes from, if he goes to that one location, cell will fire. So it's a play cell, place receptor field, play cell. Similarly, you have grid cells, which are more complicated, I won't go into. But object cells, there's an object in a particular location to see, the object should be there, then it'll fire. Then it'll fire. Sorry, it won't fire. Remove the object, it starts firing like crazy. Where's that object? I saw the object firing like crazy. So an object cell, existence of an object cell. And many miniature cells in the hippocampus, coded by time and space. And there's a band of fibers called the inferior longitudinal fasciculus, you can see that. Inferior longitudinal fasciculus goes and projects to the angular gyrus in left hemisphere. Involved in arithmetic computation, involved in sequentiality, left, right, spatial representation, all of those things you need for a calendar, interacting with the hippocampus. And I was amazed the other day when I was testing one of my subjects who had a calendar line. I showed it's a real phenomenon. Guess what happens? This borders on the supernatural almost. But of course it isn't. There's a rational explanation somewhere, but I don't know what it is. This lady had a, Rohini, my student, had a beautiful calendar in front of her, shaped like a test tube. A ribbon with a Mobius strip at one end of the ribbon. A nice ribbon dangling in front of her, straightforward ribbon dangling in front of her like a test tube. But this end is twisted to form like a Mobius strip. She didn't know that. She just knew it was slightly twisted. Then we put numbers. She asked her to imagine the calendar. She said, oh, January, February, March, April, May. It's a bit fuzzy here, doctor, until I get to December. So October, November, December, fuzzy. She didn't know it was because it was twisted. She said, look carefully, what do you see? Oh my God, doctor, the letters are mirror reversed. And of course they would be, because you see, if this ribbon is printed, if you twist it one, it's going to mirror reverse. She hadn't figured it out till I pointed it out, and she said, it's mirror reverse. But there is no ribbon in the bloody brain. There's no physical entity. So what's he talking about? Then I said, come back. You're imagining seeing it from the other side. Let's imagine you're seeing it from the other side. What happens? Who's the other side? He said, woman, now I can read everything on the side. And the other letters are mirror reverse. How is this possible? There's no homunculus looking at the brain. There are representations, symbolic descriptions of the world in the brain. There's no replicas of the external objects and events in the brain which serve no purpose. If you have a replica of a calendar, you need another little guy looking at the replica. It doesn't serve any purpose. So if that's true, then how come he's reporting all these fascinating aspects of the calendar? It only could be true if there were a real calendar in the brain. That's an enigma. Equally surprising is sometimes the calendar, you go test your friends, they're not rare, 1 in 100 people. You can do these experiments in India. 1 in 100 people have this phenomenon. You go test them carefully. Hula hoop is going behind her. 1/3 of hula hoop is behind her. I said, how can you see it then? I can't see it. I can see it in my mind's eye. You can see January, February, March, April, June. August, September is missing. It's here. Right there. And you can see the font. No, I can't see the font. I can see the positions. Then I did this absolutely crazy experiment. I put a mirror in front of her. I said, can you now see them behind you? I see them. I can see them now. There I draw my line. Okay. I don't quite believe it, but I thought it, for completion, I'd mention that she was able to see in a mirror the projected image of the calendar that was hiding behind her left shoulder. And indeed, you didn't need a mirror. If you looked over the left shoulder, you could see the calendar. It's hidden, ordinarily hidden. If I could remember things from the past, from August, she said, August is too far back, but try and remember. August. No, I can't see it. Oh, I can see August now. Yeah, my prom night. The spatial representation of memories, which is there in the history of philosophy, a venerable tradition in history of philosophy, Indian philosophy and in Western philosophy. Okay, I think I'm going to conclude with that by saying that it's been nice coming here and sharing my thoughts with you. Hope I wasn't going too fast. And we'll have a discussion, I suppose, soon after, and take some questions from one of you.
Thank you, Professor V.S. Ramachandran. Thank you very much for such a great talk. When I was in first year postdoc at NIH, I always wanted to hear your talks. And today I got to chair your session. It's a great honor for myself. I know into NBRC, I work at National Brain Research Center. I see the tree which you planted at NBRC, and I will take a picture and send it to you. It's doing well. And you need to come back again to NBRC to plant another one. So we look forward for that when this COVID crisis, we get over with that. Usually in distinguished lectures, we don't allow questions. So, but I see there are some questions which have come up. So I don't know what to do. Professor Ramachandran, what do you think? Or my co-chair, whatever he says, we'll go with that.
Whatever Professor Ramachandran wishes. If we, yeah.
Should we go?
We can't hear you.
We can't hear you now. Something happened.
But if they have no time and they want to listen, they can stay and listen and ask questions for the next 15 minutes. Whether there's another show going on or not. Is there another lecture going on now?
The next one is starting at 8 o'clock. Maybe 2 questions, probably. Yes. Yeah. A couple of questions, maybe Pankaj we can take.
Okay, just take some of them. So there was this general question about, this is a really interesting one which I found was, and that is that, is it true that we perceive the cold stimuli faster than the hot stimuli? Is the map of somatosensory cortex is more regarding the former than the latter? If you could clarify on that, sir.
Within the latency experience sensation, what are you talking about? Are you talking about latency? The cold and hot, are you saying are the different latencies to feel it? Is that what you're asking?
Okay.
Depends on what you call hot. If you touch a hot plate, It's immediate response.
Okay. I'm getting confused here.
So,
Ask the question. What is the question?
so
Yeah.
do we feel the hot and the cold at different times? Is it only because of latency, or is there something to do with the
Temperature.
arrangement of the?
That in my head.
So we can't hear.
Okay, I don't maybe.
Yeah.
What is my? You need no volume. No volume.
Yeah, your lecture was very well audible. So
Okay.
now it's the issue. Yeah. Okay.
So what I'll do, in the interest of time, Professor Ramachandran, if you would allow everybody knows your email, so we would post those questions to you. If you could find time to reply to them, that will be really appreciated. I think that will be better because we're losing the audio for some reason right now. Your talk was wonderful. We could hear you very well. We could see your slides very well. So we greatly enjoyed the talk, and thank you so much. Thank you so much for joining. I know it was hours on a Sunday morning for you, but thank you so much. We really appreciate your presence for this meeting. Thank you.
I would also like to say it was such a fascinating talk. Many thanks. And I'm always deeply impressed by your ability to use property of brain plasticity, mirror neurons, cross connectivity to treat clinical problems. That has been wonderful. And your contribution to behavioral neurology is truly awesome. Thank you so much. So we are handing over to Anita.
Oh, sorry. Okay.
Thank you.
Open then. Okay.
So thank you, Professor Ramachandran, sir, for your excellent talk. And Professor T.R. Raju, sir, and Professor Pankaj Seth, sir, for chairing the sessions. And now, before we go to the next symposium, can we have 10 minutes break?